So Sarah, you happy to start with your presentation?
Sure, yeah.
So just a quick introduction for everyone who doesn't know Sarah. Sarah is someone who is subcontracted through Afterlife. She works for C2W, and she produced a really significant part of our toolkit, which was amazing. But I won't go into vast amounts of detail because I'm sure you're going to cover that all. Take it away, Sarah.
Thank you very much. Right, I'm going to do my level best to do my screen share and then hit the magic button that allows you to see my presentation. Thank you very much for having me here to Rapid Revelations, which is a bit of a tongue twister, which has led me into a number of dark alleyways as I've been trying to describe to people what it is that I'm doing today. First of all, I would like to say, so my company is C2W Consulting, which stands for Cradle to Wave, and it was the best thing I could come up with because black consulting as I was then would have sounded rather dark, and brown consulting as I am now as a married woman also sounded rather dubious. So I went for C2W, and ever since then people have asked me what it means. So there you go, Cradle to Wave. Brought up by the sea and ended up working on the sea. So it's no surprise that I do marine biosecurity planning as a big part of the work that I do as a freelancer based on the west coast of Scotland. And I pull in a lot of different people to work with on biosecurity plans. And this particular piece of work, I did a lot of work with Chris Wood from the John Bishop Group down in the Marine Biological Association down south. So a big kudos to Chris. I believe she's on the participant list. So hopefully she's her ears are burning slightly as we go through this. And if anybody's got any technical questions about biology and species and all those kind of wet things that we find in the sea that we don't want in the sea, Chris hopefully will come on to the chat and save me. So going on, what was it that Rapid asked me to do? I got the exciting project title of 6.1 and 6.2. And the coding behind that was 6.1 was Marine Biosecurity Planning Toolkit for marine site managers, and 6.2 was a set of training packages and talks for non-specialists. And I'll go into a detail about what those both those things mean in just a moment, but it's really important to say that what we tried to do with this piece of work was to work closely with everybody else who is working on the Rapid Life project, to make sure that there was consistency between what the freshwater groups were saying, what the terrestrial groups were saying, what the marine groups were saying. That was about as easy as I know this audience will know it sounds. It's never simple to kind of translate across the different marine, terrestrial and freshwater areas. Very different challenges in different areas, but it was important to us that they were at least aligned and consistent so that we would try and minimize the confusion that can abound in these areas. It was two parts of a larger 4-part toolkit. So the other 2 parts were the freshwater and the marine and the terrestrial side of things. And the idea was to take existing materials and then add to them new and relevant detail, and then very much to shape them for specific audiences. We wanted to, under 6.1, to create this biosecurity planning toolkit. So the idea was that if you were running a port, you'd be able to see relevant material just for you, and you'd be able to pull it out and kind of take all this existing guidance and have it in one easy area to look at and to use. And again, the toolkit talks were there to provide a more sort of narrative style approach, so that it was very much aimed at a non-specialist audience, a non-technical audience, and it was supposed to sound and be a bit more hearts and minds kind of approach than the toolkit, which was a bit more business-like. I hold it a close secret, not terribly closely guarded secret, that I'm not a marine biologist. I am a facilitation and stakeholder engagement type person. I do communications by and large with marine audiences. I come from a boating background, so I'm very familiar with the recreation, and I've been a marine planner for some time as well, so I'm familiar with that side of things. So the first thing that we did, and sort of playing to my own strengths, was to go for stakeholder engagement. And again, APA and Alexia and co were very keen to make sure that we were engaging with people who were going to be the user groups at the end of the day of these tools that we were producing. So what we wanted to do was to identify those key audiences and then identify any barriers to the uptake that they might have, and to look at the kind of the information that they would need to do effective biosecurity planning going forward. We wanted their buy-in, but we didn't necessarily need them to sign off on the materials. So it wasn't that they had control, but we wanted them to help us shape the language that was being used, and to hope that the final draft would not be a surprise to any one of these people in particular. So we worked with the Royal Yachting Association, British Canoe Union, the Green Blue, British Marine, the Seabed Users Development Group, the Inshore Fisheries and Conservation Authorities in England, Shellfish Association, Angling Trusts, British Ports Association, UK Harbour Masters Association, as well as some elected members and officers in the related coastal authorities. So it was quite a big and diverse user group, and that's not actually the full extent of the list of the people we engaged with. There were quite a few others as well, but it felt daft to be putting them all on a slide and just reading you out a list. But it was very important that they were involved, and it was really quite interesting talking to them. We did a series of interviews to start with, and then we involved them in the development of the language and the tools as we went forward. And I was interested in talking to them about what their understanding of invasives were, how they would use these things, these tools that we were producing. Would they indeed use them? Was this a bit of lip service, or would they actually embed it into what they do? So there were some challenges in there talking to people. The toolkit looked like this whenever we first decided what it was that we were going to do. Once we'd had those discussions, it was the initial plan was to have a kind of website, and you can see that the landing page here is where it would start. There'd be a static text box with some key documents like the legislation and the key guidance that they might want to go and look at. There would be another static text box with the 3 golden rules, as we called them at the time: vigilance, reporting and cleaning, and then how to report your sightings. Going back to what Niall was saying earlier on, the spending in this area, I think we need to start managing expectations about what can be achieved sometimes. Whenever we looked at the cost of developing a website like this, and then who would maintain it going forward, and so on and so forth. The GB Secretariat, we're all very grateful we're there to be able to be the host at the end of the day, rather than having to develop our own website and to then worry about how that would be maintained going forward. But this was the structure, this is the kind of logic flow that you would go from the landing page, and then you would decide whether you were a large port or a small scale operator. Then you would look at managing invasives, legal situation, what other groups are doing, and so on and so forth. You can see that flow through. One of the things that really came out at this stage was that the stakeholders really desperately wanted to know, first of all, why did they have to be doing this? So the legal situation was really important. They wanted to know absolutely chapter and verse exactly where they stood in terms of legalities dealing with invasives. And the second thing that they wanted, and has always been my experience in doing biosecurity planning with communities and local groups and so on, is that they are very concerned to know what other people are doing. And I think it's a very human response, and we've seen it during the coronavirus pandemic as well. What's my neighbour doing? Why are they doing it? Are they doing the same as me? Are they putting as much effort in as I am? Does it impact them as much as it impacts me? You know, will they be disproportionately have it easier than I will for some reason? And so the final product actually ended up having quite a lot of that sort of information in it, so that people could actually compare and contrast their experience with other similar user groups. So the toolkit itself, we had a number of different sections. Some of these have now been amalgamated in order to fit onto the GB Secretariat website, either because they were already there. So for example, the landing section, what are invasive species, and that kind of thing, that's already very well covered under the more general sections of the site. So some of these have since been amalgamated, but some of them are still there. And just to highlight a few, you can see there again what I was saying earlier about this best practice by sector, different impacts on different sectors, you know, the different types of sites, so large-scale sites such as a large port or harbour, or a small-scale site such as a more local development, or a, you know, just a simple slipway or a small yacht club, something like that. What other sectors are doing about invasive species, who's taking an active look at things. But one of the interesting things, and one of the subjects that's come up a lot in other biosecurity planning work that I've been doing in the marine environment, I've highlighted their options for infrastructure developments. And this was a section where we looked at, if you were going to be doing infrastructure, so this is sort of future casting, if you were going to be doing an infrastructure development, what kind of infrastructure might be less friendly to invasives than the kind of the normal development that we might see happening. And along with, are marine biosecurity actions, mitigations effective? That's one of the big question marks that remains sort of hanging over marine biosecurity planning in particular. What kind of infrastructure might we put into the water that would be less friendly or indeed even hostile to marine invasives? And by the same ticket, you know, what kind of evidence base do we have for the decisions that we're making in terms of biosecurity planning? So I'll give you, for example, we often say in marine biosecurity planning that it would be sensible in the winter for people to drop their moorings to the seabed so that they would be smothered. They anchored the mooring chain and so on, the riser would all be smothered in the soft sediments that you often find on the seabeds underneath moorings. But if they're falling onto rocky substrates, then that might actually make the matter worse. So do we advise people to do that, or do we not? And where's the evidence base for that kind of decision? On to the training packages, 6.2, as it's snazily entitled. So these training packages were very much, the first question was, who are we targeting them at? And we looked at all sorts of marine user groups, event and regatta managers and volunteers, and should we be looking at liveaboards and people who are using their boat year round, or seasonal users, day users, and so on. Would commercial operators actually be outside of the scope? The final list there you can see on the right hand side: commercial ports and harbours, event managers, marinas and large yacht clubs, mariculture, volunteers and mooring associations. Obviously some people are going to feel left out if they looked at that list. Some people might wonder where they fit in that list. But we felt that these were the people who had an opportunity to influence how biosecurity was managed on site, and therefore they were the key user groups. This is what the slides from that particular set looks like. So for example, we've got commercial ports and harbours here, and I've just done a screen grab of all the different slides. And you can see we start with what are invasives, and then economic impact, the complexities of regulation, looking at Schedule 9 and so on, coming through risk assessment, and then we go through the biosecurity actions themselves. If we look at volunteers and mooring associations, it's a slightly different set. The tone is different. We look at more the kind of the impact on the individual. We look at the kind of loss of amenity and hull fouling and what that might mean for your boating. We do mention Schedule 9, but it's more in a light touch kind of way. And we move on quite quickly into a light touch on biosecurity planning itself. So it's not the full process that we might recommend for a commercial operator. And then we're also giving a case study. In this case, it was the Commonwealth Games flotilla, so it's something people could relate to in that context. The idea was that people would be able to give these presentations to any group, and they would tailor them, well, they can tailor them for themselves, adding in different sections as they went. So I'm just going to take you a quick whiz through the biosecurity planning process. It's a 5-step process, but we kind of tag it as being a 2-step process. So step 1 is to get senior support. The emphasis being that if your committee in your yacht club or your senior management in your port authority don't buy into it, then you really don't stand a chance of putting any of these actions into place. So get senior support. Then the second step, so 2 easy steps: get senior support, then write a plan. How simple can that be? So you're writing your plan actually breaks down into section 1. You're looking at an introduction, why you're writing the plan, usual bit of preamble as to what it all looks like. Section 2 is the scene setting, so it's trying to get people, it's basically scoping what area is going to be covered, what the time limits, what activities are in, what activities are out, and looking at useful contacts. And you'll see these are just screen grabs from the presentation. So we put in example pictures, but we've said to people, choose an image relevant to your audience. So all of these things were to be tailored. The third section then is about the environmental information. So we're looking at the physical structure, water chemistry, and the invasive species known to be in the area or coming your way, and designations and that kind of thing. Before moving on to how is the area used, how many boats, what type, where are they coming from, what stock movements there might be, and the level of complexity will depend on the person doing or the organisation doing the biosecurity planning. And finally, these are the actions. So there was a great long list of actions, and if you look on the GB Secretariat website, we've got quite a long list of a sort of smorgasbord of biosecurity actions. And again, I draw your attention to the lack of data on a lot of these things to prove one way or the other how effective they are in different contexts in particular. But communications is a big part of it, reporting and having that open culture of reporting and encouraging people to actually say what they see is a really important part of biosecurity planning. And it's one of the interesting things as a contractor, I find a lot, I actually get quite a lot of communication from people that I've worked with over the years saying, I've just seen X. Do you think it's one of these things that we talked about 3 years ago? And I find myself examining these pictures and then saying, well, have you actually spoken to your manager? Oh, no, I don't want to bother him, her with that. And it's reinforcing that you need to be reporting these things in-house. And the final step then is the monitoring and review of the whole process and keeping that iterative process going so that things get monitored and people come back to the start and make sure that the plan is still active, that there is still senior support, and so on and so on. So this process has been going and been recommended by Natural England and CFAS and SNH and so on, NRW and so on. For a number of years now, and it seems to be bedding in and people seem to be following it. When I see CalMac's biosecurity plan or kind of the big port operators, they're following the same process, and it's good to see that this is something that seems to be chiming with people. It fits in with the standard kind of process for risk assessment and for the HACCP kind of for the health and safety hazard analysis critical control pathway process. And so there's an overlap between those two things to make sure that it's a familiar process. Throughout the 6.1 and 6.2, we found that there were things that people were asking for us time and again. They were either part of one thing, part of 6.1 or part of 6.2. So for example, the horizon scanning species lists. So those were done for the English coasts. And that's available on the website. Now that's a couple of years old, but again, that was the MBA, did that excellent work. And that in itself has proved to be a very valuable resource. We also produced a lot of additional pictures that demonstrate impacts, and we did this lovely standalone 6-minute animation, which I'm particularly proud of. And I'm going to ask the organisers, Alexia, do we have time to show even a little sample of the animation? I think maybe just show the first couple of minutes. Hello, hello. I'm going to show the first couple of minutes.
Fine. Sorry, yes, that's fine. I thought I'd unmuted myself, but I hadn't.
Cool. Hopefully everybody can hear this.
I don't think we can hear the sound.
Can you not?
If you have it anywhere else, if you can play it through another application and then share your screen, that sometimes works.
Okay, let me see if I can do that. Nope. I knew there'd be a technical challenge somewhere along the line. So if
yeah, don't worry. I feel your pain.
Are you now seeing my YouTube?
No, you'll have to change the screen that you're sharing.
Yeah.
So if you then share your YouTube screen, hopefully we'll be able to hear that.
That's the one. Thank you for that guidance.
I still can't hear it.
No. What a pain.
Okay, well,
Because
I can
because
hear
of
it.
the
There's some beautiful, beautiful music, and then a lovely
you get the
voiceover.
idea from it.
You get the general gist of it. So it is, if I just scroll through a little bit here, it is a little bit entertaining, a little bit tongue-in-cheek. And I know that some people looked at it and thought, oh, God, you know, aliens and, you know, it's all a bit silly and how big can an invasive species really be and all this kind of thing. But actually, it really chimed with a lot of different audiences, and people got the gist of exactly what it was we were talking about and the concerns that we might have about invasive species in 6 minutes, in a nice kind of package, which allowed people to really engage with the topic.
I'm just going to put the link up here on the chat so that people can see it.
Cool.
Yeah. What
I wanted to do was also, I thought right at the end we definitely don't want to grow our own coral. Not today anyway. What I wanted to do was to say thank you to everybody that was part of the whole. Oh my goodness! I've now got a woman growing coral in my ears. There we go. I wanted to say thank you to everybody that was part of that program, because it really was very much a team effort between the AQUA and Natural England and MBA and myself. So I think that's probably my time just about up, and I wondered if there were any questions.
Let me just see if I've got any. Hang on. I haven't got any yet. I'm just going to put the link up for the video for the animation. It is really good. If anyone has chance to watch it, I really recommend it. It's quite a good one for introducing people to why INS are important. And yes, someone's asking if it's suitable for children. I would say yes.
Yeah, it's not that scary. Yes,
No.
Absolutely. We tried to keep the language very much suitable for all ages, and I have showed it to very young audiences, and I've showed it to commercial divers, and I've showed it to port authorities. I've worked a lot with ABP ports on their biosecurity management, and they all really like it. They all really like it. It just makes the point in a very gentle way. And I can be so flattering about it because I didn't write all of the script. It was Kyla that I was working with on doing that, and Kyla Orr, and she's an excellent science communicator, and she just knocked the edges off some of the language that we were using.
Yeah. Sorry, I'm just checking if I've had any more questions. I don't think I've had any more come through. Jen, have you got any there?
No, I haven't got any private ones come through. Just a comment that it was a great talk.
Cool. Someone just said also, thank you. I'm targeting Girl Guides and rainbows, so this looks like a perfect introduction to invasives.
Excellent, excellent. Really glad. Enjoy.
Well, thanks very much for that talk, Sarah. That was fantastic, and I can't believe how much work you and put in. Yeah, I know you had a lot of input and help, but still to the IS management toolkit, it really is amazing.
Thank you very much.
Set of tools.
Cool.
And I think we're going to have to move on to our next presentation just because of time. You've actually helped us manage to catch up on time, so thank you, Sarah. So I'd like to welcome Andrea Griffiths. Oh, sorry, Jen even. I lie. I'd like to welcome Jen Nightingale. I can't read my own agenda, apparently. So I think Jen doesn't really need much introduction. She works for Bristol Zoological Society. She is what I like to call the crayfish queen. And she has been absolutely instrumental in the Rapid Life project and has been leading on the crayfish aspects of the project, as well as the waterbodies accreditation scheme, AQUA. And I will let her sort of tell you a bit more about it. Take it away, Jen.
Thank you. Can you see my screen okay? Is that all good?
Yep, it's working.
Great. Okay, so good afternoon and thank you to everybody for sticking with us. It's been really informative so far. So I'm not sure if I am a crayfish queen. I think I've progressed from a toad lady. Is that a promotion? I'm not sure. And so I'm the UK Conservation Manager at the Bristol Zoological Society. And I'm going to focus today on the work that we've been doing for Rapid Life. But I thought at first I should probably introduce the subject that I'm talking about. So this is the white-clawed crayfish. It's our only native species of crustacean, crayfish crustacean. So it's freshwater, related to a lobster, and it's slow growing and it has a relatively slow maturation. So it can take between 3 and 4 years to actually start breeding in the wild. And it only breeds once a year, and the females only have up to maybe 70 to 90 eggs and hatchlings a year. So it can be, and only about 10% of them go through to the maturation point. So you can see it takes a while for them to establish. They're very flexible. They can live in both river systems and lakes. And they eat pretty much anything. We actually term them ecosystem engineers because they're really useful at breaking down detritus and cleaning up the environment. And they're actually a keystone species, which means if you take them out, negative things happen. So when we started this is the status of them here at the moment. They're throughout not just the UK, England and Ireland, but they're also throughout Europe. However, they're globally endangered. Their status increased to endangered by the IUCN in 2010. Now, when we started this project back in the day in 2008, we mapped the Southwest and found there were only 18 populations left. So there'd been a 70% decline. And you can see from these maps that there's a problem here. And that's mainly to do with invasive species, the signal crayfish. And a little bit more on that now. So the decline of the reasons for decline, the usual suspects, things like water pollution, habitat fragmentation. Coral, they're very, very sensitive to things like high nitrates and phosphate loadings. And the main problem for them is invasive crayfish species. Now, the key one that we have in the United Kingdom is the signal crayfish. This was brought over in the 1970s to be farmed. Very much a similar story to the American mink, which was brought over for the pelt. We brought this guy over for the food industry. Ironically, we mainly exported it to the Scandinavian markets. But the problem with the signal crayfish is it carries something called crayfish plague. Now, this is kind of like a mold that infects our white-clawed crayfish, and at the moment it will kill entire populations within 2 weeks. Not only that, even if it doesn't have plague, it's a bigger aggressive species, so it will outcompete, it will predate on ours, and it will basically take over an environment. Sometimes when it gets to very high carrying capacity, it can cause these biological deserts. It basically eats everything in the environment. Now, there's also financial implications for this. It can cause flooding in lowland areas because it actually creates very extensive burrows within banks. So you can see how this can cause flooding. It will also predate fish, so it can be really debilitating to things like our salmonid industry as well. So it's really bad news, and unfortunately it doesn't just stop with signal crayfish, but we have other species now in the UK. Now, all of these species that you can see here, these are all species from North America. So although the populations are relatively localized in the UK at the moment, they can all carry plague. So potentially they are a very serious threat to our species. And I should say they can all breed. They're temperate species, so they can breed in our waters as well. Now, the other species that we have in the UK, we've got the noble and narrow-clawed. These two are both European species, so they're actually endangered as well from the other invasive species in their native habitats. And we have localized populations, but these guys don't really cause any issue to our crayfish. It's just to make you aware that you could actually see them. The red claw in the middle, this one is the only species allowed in the aquarium trade. So this is a tropical species, and that's allowed. But probably the one of the most serious threat is the marble crayfish down at the bottom here. This one is a temperate parthenogenic species. So if it gets released, it only takes one crayfish potentially to have a population. Now, although it's not in the UK waters at the moment, it is being found in Europe. So it is a particular concern, this one. And again, it's illegal to have it. There are some in the aquarium trade, but they're not allowed in the UK. So what did we do to try and stop this? Well, we set up the Southwest Crayfish Partnership in 2008. This was a group of professionals such as us at the Bristol Zoological Society, the Environment Agency, Wildlife Trust, CFAS, Buglife, and associated crayfish professionals. And we set it up with the backing of the Natural England Countdown 2010 funding. And we set up 5 key strands of the project. And these ones are pretty much, they've carried on throughout. We are mapping counties to work out not only where our native crayfish species are, but also where the invasions are. We're establishing ARC sites, safe sites. We're doing outreach communication. We've heard a lot about check, clean, dry, really promoting the messages that go and enjoy your rivers, but don't forget to check, clean and dry your boots, your gear, between things. Not only can you spread invasive plant species, but you can also spread not just invasive crayfish species, but also crayfish plague, which can actually stay dormant within damp conditions for up to 2 weeks, and then reinfect populations if you accidentally then use your dirty wellies in a new place. So really promoting this. We've also been doing our captive breeding and piloting invasive control studies. So we're now on the 3rd phase of the Southwest Crayfish Partnership. So what was our plan for RAPID? Why did we become an associate partner? Well, we had several key targets. I haven't given them their labels like Sarah did. I'm not quite sure which number and digit we're on. But basically, we have 4 key targets. ARC sites, captive breeding, invasive control, and the Biosecurity Accreditation Scheme. And I'm going to touch on 3 of these today and leave the Biosecurity Accreditation Scheme, leave that to my partners in Southwest Water and the Southwest Lakes Trust. So what was our target with ARC site establishment? Well, very simply, I say, tongue-in-cheek, to set up 3 ARC sites during the duration of the past 3 years. So an ARC site, what is that? Well, basically, it's a safe site. So it can be a river or it can be a still water site, something that is potentially free from invasion over the foreseeable future. It's got to hold water, have the right water quality conditions, and be somehow discrete and isolated from potential invasion within a river catchment. So when we're looking at an ARC site, we do a lot of sampling, a lot of surveying, and it can take up to 2 years to actually get an ARC site from finding something, thinking it's a good idea, to actually getting the animals in it. So we do water sampling, we survey not just for crayfish species. We can't put in animals where there are already crayfish present. We want to know what's there in terms of invertebrates, newts, what plants are there. Of course, if there's something of notable concern or a priority species, we wouldn't put crayfish in there either. We also need to know that the landowners are on board, that the land use around it isn't going to change in the foreseeable future. And we monitor things like temperature and climate proofing. Is it going to stay viable, and is it going to overheat in that time? You're kind of trying to future-proof and look into kind of your crystal ball to see if it's going to be safe over time. Now, once we have a site and it's ticked many, many, many criteria of lots of different stages of assessment, at that point we are ready to bring in crayfish. Now, the crayfish can be either from donor populations that are in the wild, or they can be captive bred. So when we started the population, as I say, we were down to only 18 populations, and we did a traffic light system, red, orange, green. Red meant that they were very, very of high concern, highly threatened populations, usually because there are signal crayfish actually within the catchment and really, really close to them, maybe within a kilometre or so of the actual population. So in this case, what we've done, we've actually moved 8 parts of 8 threatened populations into ARC sites. Now, when we're pricking them up, we basically trap out, and we'll move up to 200 to 500 animals over a staggered amount of time into an ARC site. As I say, we can also use captive breeding for ARC sites, and this might be possible, or this might be appropriate when you've got very, very limited population left. Now, we never move a whole population, only part of a population, just in case invasions don't happen in the future. So what do we do with RAPID? Oh, sorry, to say that when we do our ARC sites release, we actually pit tag all of our crayfish. Now this is a passive integrated transponder, a little chip inside a glass case. This is exactly the same thing as your dog or your cat has, and we inject it underneath the ventral abdomen surface. It goes into the muscle layer and it sits there nicely. Now, just to reassure you, I spent a year assessing survival, growth rate and breeding successes once they were tagged, and it's absolutely fine, they're safe. So we have a minimum size that we can tag, because obviously if you're too small, you can't be tagged. But what this does, it's an incredibly powerful technique, because it means that basically we can track animals long term. We can see when a population is recruiting, i.e. if there's a crayfish without a tag, we know it was born in the wild, and we can see how far they go and how long lived. So it's really, really useful. So everything that we release has now got its own unique little tag on it. So with the Southwest Crayfish Partnership, we've established 18 sites over 6 counties, and as I say, we've moved 8 threatened populations. We've got captive bred sites, and we've also got river supplementations. This is where we've added basically crayfish up to where higher than a native population where they've become extinct. And so far, now it can take a very long time to see these guys to actually, you can imagine if you put a few crayfish into a large area, it might be very hard to find them again. So it can take up to 10 years to actually find them and to know that your population is established. So, but we're very happy to report that we've got over 55% of the sites are recruiting and very, very stable. So what about for rapid? Well, basically we had a target of 3 ARC sites to set up. And since the project began, we've actually released 900 white claw crayfish into 4 ARC sites. We've released in Hampshire, so we've released 515 over 2017 to 19. And this is basically a river supplementation where they've basically become extinct. So we've supplemented and put them higher up to a natural range. We've supplemented an existing ARC site at Sparshot College in their lake. I'll pretty much use anyone's water, as long as you haven't got predatory fish like perch or carp. I'll put crayfish in there for you, don't you worry. We've set one up with project partners Puxton Park, that's a leisure attraction nearby in Somerset, and we released 204 of those last year. And yesterday, in fact, hot off the press, we actually released 70 captive bred ones from my shed into a Somerset River. Now, I can't give you the location of that because we tend to keep them secret where they're public places. And I'm very pleased to report that a 5th one will be coming online at our sister zoo at Wild Place Project in the autumn of this year. And these are yesterday's photographs. So this is us, this is the lovely Neil Green, who you've already heard speaking today, and Holly Thompson, my Crayfish Hatchery manager. So this is us releasing yesterday. And it was just a lovely moment because it takes a long time, as I say, to set up an ARC site. So what about moving on to our second target for rapid? This is captive breeding. So our target here was to hatch over 1,000 crayfish. So what did we do? Well, just to first introduce you to our hatchery, we have our hatchery, our main hatchery is at Bristol Zoo Gardens. And that is basically, we set it up at the beginning of the Southwest Crayfish Partnership, so over 10 years ago now. And it basically acts as an ARC site. Why is that? Because it's not on a river, it's not on flow through, it's basically on treated mains water. And we have 2 systems. So we have a big outside 4,000 litre system that's got 13 tanks on a recirculation. So we have to filter it, and we basically have to chill and heat the water. And we kind of mimic river temperature as much as possible because we're trying to get these guys ready for release. We also want to breed animals in there. And the animals actually breed in autumn. So we need to drop the temperatures again, because the trigger for breeding is lower water temperatures and also reduced daylight. So we're trying to mimic as much as possible the environment to get them to breed. Now, once they breed, which is in autumn time, then when the females have eggs, we'll separate the males off from them and keep the females in these outdoor tanks. Then when they're near close to hatching, which is around springtime, we bring the mothers, the females with their eggs, into these smaller systems. These are 1,000 litre systems, and it means that we can keep an eye on the females. And it's always really exciting when the eggs have got little eyes and you know they're about to hatch. And it's a whole year of, it takes a whole year basically for a cycle. So we do get super excited about this. And so here's our model. So basically what we do is we either bring in females in berry with eggs in spring, and then we hatch them out, rear the juveniles, and health screen the female adults and then release them. Or we do, as I say, the whole captive breeding, where we select wild caught broodstock and then mate them and breed them in captivity. And this is a little corral. This is Martin Frayling, who basically taught me my craft. He was at the Environment Agency. And these are basically little pots where we put the crayfish, individual females, so that we can carry her safely to the zoo without her getting stressed and losing her eggs. And this is just when they hatch. They look like teeny tiny crayfish, except they don't have fully formed tails. So they'll stay on mum for up to about 3 weeks, and they'll have 2 molts. And then we call them stage 2. This is when they're basically ready to leave. And at this point, she predates them if we don't take her out quickly enough. So it's a real, it's a little bit edgy, this part of the life cycle. Now we've had to expand our hatchery, so I've now got them at my shed as well. So this is my house and this is my shed. I would take you on a Zoom, but I don't think reception's gonna, I'm not that confident with it yet. And I've also been running my PhD thesis out of the shed, optimizing aquaculture techniques for how to breed a crayfish, just add some water really. That's the basic kind of conclusion I've come to. And we've been extending also to Bristol Water. So this is our 3rd hatchery space where I grow on my crayfish for release. So a big shout out to Bristol Water for allowing me to steal their trout tanks for my crayfish system as well. Now, what was our output? So our target was to produce 1,000 crayfish, and I'm delighted to say we've produced 1,100 over the course of this project. At the moment, I have about 250 crayfish in my shed, and more on that in a little bit. So what was our next target? So our next program target for RAPID was invasive crayfish control. Now we had a target of having 2 control sites and reducing the crayfish population by 75%. So this is the first time that we've done crayfish control. And basically, in terms of, just to give you a quick background on crayfish control, there's been a variety of lab-based control trials over the years, and this is basically stemmed from Paul Stebbing's work at CFAS, looking at things like male sterilization and developing crustacean-specific bait matrices. Now, this has been expanded into the wild. The first project, the first field project, was with Nikki Green, and she's still continuing. Nikki Green's a professional ecologist, and she works on the River Barle, looking at male sterilization plus trapping. Now, there are other elements that you can do, for instance, trapping plus adding predatory fish, dewatering, or using biocides. Now, at the moment, biocides are the only really 100% eradication control. The rest of these elements are good for reduction, but not for eradication. Now, obviously, using a biocide in many situations is inappropriate. So what the aim of this is to stop the spread rather than eradicate completely. I would like to say here that there's a lot of great scientists out there working on further methods. So there may be a potential for full eradication without using something as debilitating as biocide in the future. So our control trial was at Chew Magna Reservoir and at the Wimford Brook. So we used a process of trapping out, and this is Neil, again, harder action. He's all over the place. He's a jack of all trades, this man. And here he is with one of our artificial refuge traps. And just to give you an idea of how this looks, so basically this is the reservoir, and the red dots are all the artificial refuge traps that we put in. So we've got a line down the middle and traps all around the edges. And basically we check the traps once a fortnight. And now the plan is to put in predatory perch as well, recognizing that when you trap, you're basically trapping out the big guys. And most of the way a population is built up is basically there's a lot of younger animals and not that many adults. So to get the younger animals, you want to bring in a predator. Now we would have brought in, we would have introduced the predator in the winter of last year, but unfortunately COVID has prevented this. So we can't actually introduce our predator until this autumn. However, we will be continuing this trial because we want to see what the trapping is plus the perch. Now we've seen some really interesting migration in and out as well from the Wimford Brook. So at the moment, I would say we've actually trapped out over 22,000 animals. Is this a 75% reduction? Well, at the moment, I would say I have absolutely no idea, because as I say, we haven't put our predators in, but we are getting a really good idea of how they utilize the site. The other element of this was our dewatering. So we had to do an estimate of 75% removal. Now this is the top of the Wimford Brook where we diverted the water, and you can see we've pretty much emptied it entirely. Now, although there will still be some crayfish within it, we're confident that we removed at least 75% of the animals there. And then the other part of the dewatering was on a concrete basin. Now this, I can assure you, this was 100% removal because we removed everything. So a man-made structure is much more effective in terms of dewatering because you can empty it completely. And with that, with the dewatering, obviously came fish removal as well. So what we did is we actually had these large containers at the side and pulled out the fish and then tracted them into the brook, so to make sure that all the fish survived during this process. And the signal crayfish were humanely killed and then basically cooked in hot water to make sure that plague wasn't still present, if they have plague, and then they were buried on site to prevent any biosecurity issue. So what were our targets? Our targets were basically 2 sites. So the tube magnet, as I said, we've removed over 22,000 animals, including eggs and hatchlings, and we're bringing the perch in autumn. And we're going to continue this until at least 2022. Bristol Water are going to sponsor this element so that we can continue this trial, because we really want to get a very good handle on it. And then in terms of the dewatering, again, very successful in a man-made structure. So definitely a recommended technique, but the Environment Agency needs to obviously be consulted in case of any flooding risks. So the other part was the Biosecurity Accreditation Scheme, and I'm going to leave that to the very, very capable Kate Hills and Nicola Morris to discuss more of that with you. But just to say, yeah, that's been something that we've been ticking along as part of RAPID as well. So just to tell you about what happens, what's the afterlife for this project? Well, it just carries on, really. We've been doing crayfish for 10 years and we're not really going to stop. So we're carrying on with our surveying, our ARC site establishment and our monitoring. We basically monitor, at the moment we're monitoring 12 sites every other year, so biennial monitoring. We're carrying on our captive breeding. We offer a lot of practitioner support and training. We do a lot of outreach. This is the first presentation that I've done in many, many years without showing you our crayfish costumes, but we do have those available for hire or just to have. And we have, basically, we're about to employ a biosecurity conservation officer, basically really working on carrying on a lot of the invasive weed control and invasive crayfish control and outreach in the southwest. Plus lots of research. I'm finally going to be writing up a lot of this and producing a crayfish handbook in case you would like to read something like that. So a massive shout out here for all our project partners, particularly the Southwest Crayfish Partnership and my team at the zoo. I couldn't do it without the likes of Holly and Neil. And a massive thank you to all our project partners as well. Now, I just wanted to say, and I know I've got a little, have I got a bit of time, Alexia? Just say yes. Just do a thumbs up. Thumbs up, because so as part of it, if you have been at Bristol Zoo Gardens, we were very, very excited to host you today. And what we would have done is show you have a tank of crayfish. So I thought, well, in the absence of being able to see a tank of crayfish, I would bring the crayfish to you. So I'm going to stop sharing my screen, and then if you put me on large in your screens, hang on, this may or may not work, I'm going to attempt to show you, let you meet some crayfish. So I'll stop sharing, and then, right, so, am I in big, Alexia?
So if you click speaker view participants, then Jen's screen should go big.
I've also got to just cover my keyboard, because the last time I did this, my keyboard got really soggy. Okay, so here we are. So this one, she's a freshly molted female. So she's captive bred, she's in my shed. And she is 4 years old now. So she didn't make it to the ARC site yesterday because she's just molted. So I haven't tagged her yet. So she'll be tagged on here, sort of just injected there. and then she will be released quite soon, actually, because she's wondering where all her tank mates have gone. So that's a female, and she's got this big, oh, she's jackknifing now. She's got this big wide tail where she basically has her eggs, and she tucks them up under here. Now, hang on, that's not all. So there's a big female. So as I say, she's 4 years old. And then we've got here, so then hang on, bear with me. So I just want to give you a size difference. So then this little chap, now that one's a year old. So he's too small to tag, so he hasn't gone out yet either. So he's going to be a couple more molts, and then he'll be tagged. Now for the male sterilization that I talked about, and sorry, I hadn't really got time to talk about it too closely in detail, but can you see, I don't know if you can see that. Can you see he's got two little sort of white clusper bits? Can you see that? And they're basically, yeah? And they're basically snipped off during male sterilization. So the big guys who hold the territories, what they do is they basically, they can't mate effectively with the females. So they hold these territories, they prevent the females mating, from having fertilized eggs, and they prevent the younger males from mating, and they can't actually put the spermatophores in the right place. So there he is, he's waving at you all and saying, really sorry you didn't come to Bristol Zoo and see me in the flesh, as it were. There we go. And then one more. Have I got time for one more, Alexia?
Yeah, you've got time for one more, and then there's some questions as well. I've got a lot of questions for you. So.
Okay, hang on. Oh, goodness. This is the hardest one, sorry. Can you just talk for a minute, Alexia?
Yeah, sure. Someone just said to me, a great talk from the Crayfish Queen. That's your name now, I'm sorry.
Okay, hang on.
Is it a really diddly one?
Oh, shush, don't spoil it. Oh, God, I really am going to drop it. I'm going to really make my... Okay, hang on. 2 ticks, please, listeners. Right. Can you...
Oh, yeah, we can just about see it.
Can you see it?
It's a little blurry, but we can see it. Is
that better?
Wow, yeah.
Okay, so this is 6 weeks old. Can you see that nicely?
Yeah, we can see it.
So that's just at 6 weeks. Oh.
I never thought I'd find myself going, oh.
Well, this is what outreach is about, Sarah. It's all about making you love crayfish.
Seeing is believing. Unfortunately, all of my samples, I think I've got the largest private collection of invasive non-native species from the marine environment, and I've got a shelf behind me of all sorts of revolting things that I could pull out of alcohol and show you, but none of them would make you go, oh. They all make you go, ugh.
Well, you see, I'm up against gorillas. That's my problem at the zoo. So the challenge is on to make people really love and care about crayfish. And as I say, there's probably about sort of 200 plus of those in the shed at the moment. And so that one, yeah, that's going to molt again. But yeah, it takes in captivity they'll take we can actually breed them in the 2nd year, so it's a much quicker process because they've got more stable environments and a lot more food. And at this size they're basically eating live food, so I'm feeding it lots of different types of shrimps, live shrimps, and gut loading them, and then weaning them onto kind of a plankton diet later. And then they start eating like superfoods like chard and cabbage, and they start eating plants as well. Right, okay, I'll do some questions and then I'll show you.
We've got some quick fire questions. Everyone's very excited to see your crayfish, by the way.
Okay.
Definitely the highlight, I think. Stole the show. So regarding arc relocation for white claw crayfish, are they quarantined prior to release, especially if they're sourced from a river that has signals?
So if they've got signals in the river, then they would be health screened in the river. So basically if they've got, so signal crayfish can either carry plague or not, and the Environment Agency usually tries to screen them periodically so they know whether they are plague carriers or not. So if we were going to do something from that sort of case, we would have to health screen a large proportion of the white claws to make sure that they weren't carrying plague. And in that situation, they wouldn't go into another river because it would be too dangerous. So in that situation, they would go into a closed water body. I mean, all crayfish are screened before moving. So in a case of if you're moving, say, 500 animals, you can't bring them all into captivity and quarantine. So what you do is you do a proportion of them, you health screen them before you move them. In the case of the captive breeding ones, what we do is we sacrifice a percentage, and that's really hard to do. So this year I had to sort of select an amount of crayfish to basically go for health screening, and it's a one-way ticket. It's not like when we do a waterfowl reintroduction and we can just do some skin swabs and an x-ray. We basically have to kill the animal, so then it's awful. You have to kind of choose which ones to select for screening, but it's really the only way to make sure that you're not bringing in novel disease. And the other thing I would say is that when you are moving a population after health screening, we try and make sure it's within the same river catchment, so that you're not bringing in anything potentially into a new river catchment, if that makes sense. And the majority of our ARC sites these days are still water sites, which are kind of standalone water bodies.
All right, I'm going to have to quickly rush to the last few questions before we move on to the next presentation. Minimum size water body which could be considered for an ARC site?
Probably something like a 2 hectare, yeah, probably I'd say 2 hectare lake, something like that. Possibly 1 hectare, but it would be more like a stocking pond. So we'd use it as a harvesting pond. So you can have smaller ones, but they're harvested more than obviously if you want the larger the better, really. Yeah, for an ARC.
That makes sense. From Zoe Trent at Southern Water, do you have any, sorry, I haven't said the names of everyone else. Do you have any concerns about genetic diversity? Does the remaining UK population have enough genetic diversity to be resilient?
So it's pretty poor. There's a bit more genetic diversity up north than there is down south. So wherever possible, we try and bring in quite a large proportion of males and females. So we try and kind of have about sort of 300 as a bare minimum for an ARC. So yesterday's 70, they will be increased. We'll be supplementing them for the several years to get a kind of as diverse we can. But in reality, none of the populations are very diverse. So we're not too concerned about this. And what we're on the hunt for at the moment is populations that are resistant to plague, because there are some populations of white claws in Europe which are plague resistant. So we're trying to find some of them here so that we can then breed that resistance. And there's some really clever genetic scientists looking at the kind of at molecular level, what makes a crayfish resistant to plague. So more on that to follow. That's the next phase of my project, is to breed amazing bulletproof crayfish.
Right. Last 2 questions, and then we better get moving. What do you find is the most effective predatory fish species?
Perch. Because perch are little and they're wily, so when they're little they can get into refuges. They're very tenacious, they're very voracious feeders, and usually they're found within the same catchments as well, so it's not like you're bringing in anything novel. Any animals, so for instance with the true magna one, it will be locally sourced and health screened animals obviously that we put in, but perch is a great predator. There is some work happening in the north with eels as well. But eels, because of their endangered status, it's much harder to utilize them, and they're in less supply. The supply of them is less. Okay, one more question. I'm trying to do quick answers, and it's fine.
They're very good answers. Also, someone else is just saying they attended your crayfish course and that they love the crayfish and they can't recommend the course enough.
Thank you.
So
we
if anyone
didn't
else
do it
is
this year
interested,
because of COVID, but it'll be on again next July.
Yeah, so if anyone else is more interested in learning more about crayfish, that's the place to go. So the last question was, would people be able to come and see the work in person?
Yes. So we do, yeah, we have a lot of, obviously a lot of work at Bristol Zoo. So people are more than welcome to come and visit me at Bristol Zoo, and I can show them around the hatchery. And we do have, we do allow people to come on some away days as well. So basically just contact me. Sorry, I forgot my last slide had my contact details on it, but
we'll circulate all the contact details for the presenters anyway.
But yes, there are opportunities to see the project in working.
Fabulous. Well, I think we'll have to stop there, I'm afraid. But that was a brilliant talk. We really love your enthusiasm. And now we will be moving on to Andrea, who's going to talk about the Southeast Regional Invasive Alien Species Management Plan, so the Southeast RIMP.
Okay.
And just a quick introduction to Andrea. Andrea is works for the, I can't remember it now, Medway Valley Countryside Partnership. think that's right, and is one of our subcontractors who's been really engaged with Rapid and worked on several of our contracts. And yeah, she's very enthusiastic about INS.
Okay, thank you. Right, share screen, sorry, hang on. Right, hopefully that's working. Can you see that okay? Yeah?
That's fine, yeah, that's working.
Okay, lovely. Okay, thank you for that. Not sure I can actually follow that though, Jen. That was amazing. I'm gutted to actually go next because this is going to be as dull as ditch water in comparison. But I shall do my best to keep people awake following your amazing talk. So thank you for that. Yeah, so I'm Andrea. I work for the Medway Valley Countryside Partnership. So this is us. We do loads of different things where our invasive species work is classed as one of the local action groups, but we do loads of other things as well around Kent, specifically in Tunbridge and Malling and Maidstone boroughs. So we work to protect and enhance the local environment and green spaces and engage with people and local communities, etc. So we do loads of different projects. So you can obviously have a look at our website if you want to know more about what we do. With regards to invasives, we go beyond our borough work to cover the whole of the Medway catchment. So it's a huge project. We cover the whole of the catchment from East Sussex and parts of Surrey right up to the estuary of the Medway. So it's quite a large project, and we're looking at predominantly 5 different species of invasive plant. So giant hogweed, Japanese knotweed, Himalayan balsam, floating pennywort and water fern predominantly, but we will do other things as well. So this is a picture of me. You can just about see me looking rather bonkers, hiding behind a huge forest of giant hogweed, probably breaking some health and safety rules there actually, to be fair. But yeah, so giant hogweed work, we've just been doing loads and loads of that, obviously, this time of year. So despite social distancing requirements recently, we've just more or less completed our giant hogweed work for 2020, and we've treated over 14,000 plants so far this year on the catchment, so that's great. We do loads of awareness raising as well. So we've written a children's book to help raise awareness with children. It's about sort of riverbank management, but it goes into the different plant species as well. So that's been really nice, and that was given out to loads of local schools. I do a lot of public speaking, so this was us at the Property Care Association Weed Conference a few years ago. And as I say, we do a lot of work with children, so this is one of our volunteers pulling parrot's feather, I think this was, from the school pond. So loads of different work like that as well as our main catchment control work. So we've been lucky enough to be involved with various Rapid Life projects. So we've been working with CABI to look at the biocontrol trials for Japanese knotweed and Himalayan balsam. And I'll let Corinne and Co tell you about that obviously shortly. We've been doing floating pennywort management as part of one of the small scale control works, which has been fantastic. We ran some of the priority species workshops, which were brilliant, really enjoyed doing those, that was really good. And we wrote the Southeast Regional Invasive Alien Species Management Plan, or the Southeast RIP. So it's that I will now talk to you about specifically. So Alexia introduced the Southeast, or the RIMPs, earlier on when she talked about Rapid. So there's a RIMP for each of the 5 regions of England. And the idea behind these, as Alexia said, is to bridge the gap essentially between the high level strategies and the work that's going on the ground with local action groups and the like. And we wrote the Southeast RIMP one, so you can, the Southeast region, so you can see the different regions there. We wrote the Southeast RIMP with the Medway Swale Estuary Partnership. So we're the Medway Valley Partnership, so we tend to work along the freshwater Medway, but we partnered up with the Medway Swale Estuary Partnership, and they concentrated on collating the information for the coastal and estuarine areas, where we obviously concentrated on the freshwater and terrestrial. So hats off to Medway Swale, they were great partners in this. And this was the Southeast region, so not the biggest of the regions in England, but certainly had its complications. What I did was I broke it down into catchments as well, so I didn't want to do an overarching southeast regional document. I wanted it to be very catchment relevant, so that people who might come into a new job, say for example in a catchment, would be able to get very specific, very local information. So we broke it down like this. So this was the southeast region. And what we did, we spent an awful lot of time collating historical data where we could, to look at what has obviously been recorded where previously. We contacted a huge amount of people in the southeast and made some great new contacts. And part of the RIMP was to also show who was where and what kind of organisations people might want to discuss this with in the future. So that was really helpful, so that someone else coming along in the future won't have to start from scratch. So we made loads of new contacts, and loads of those people gave us lots of information about data with regards to species presence and absence. So that was really good. And we did lots of obviously map research, trying to look at where hotspot areas might be and where high risk areas for future invasion might be as well. So a huge amount of background work obviously went into this. We held a couple of consultation events in Kent, and we also had additional meetings with various stakeholders in Kent and London and in Hampshire as well, which was lovely. And we obviously shared the draft version around as much as we could before it went live. We did come across various problems. Firstly, time. It took a lot more time than we thought it would. A lot more work than we thought it would initially to write, which we didn't really factor in. And we didn't factor in things like the cost for data. That we found the cost of some historic data quite astronomical, and that hadn't been factored in. So we did have to rely a lot on kind of word of mouth and what local people on the ground said was where, which was great really, because you need that local knowledge. So that was really nice. But the fact that the time frames meant that we were collating all of this and writing it during the spring and summer didn't really help, because some people that had a lot of knowledge perhaps couldn't actually feed in that much because they were so busy during the spring and the summer. So we did have some problems, but overall we were really pleased. We ended up with a huge amount of data on a catchment by catchment scale, but that in itself caused problems in the fact that my first draft was about 800 pages long. And Alexia was like, what? So I did have to spend quite a lot of time trying to salvage what we'd actually achieved, but obviously cutting that down massively into being a more workable document, which I think we did. I'm quite pleased with it. The end result, which is now obviously along with all the other RIMPs, you can download for free from the Rapid Life website, or the NNSS website, the Rapid Life section of that website. So the final document has a lot of information about species, obviously, what's present, what's not present, pathways, hotspot areas, areas of high risk, and obviously each individual species has got information on it. There's loads of information about pathways as well, potential pathways into the region and all that kind of thing. So quite a lot of information that is in there. So as I say, each individual species has a kind of row in these various spreadsheets telling you about its risks, the impacts it would have, and also lots of links to other areas where you can get more information. And there's lots of information on there about high concern species or blacklist species, so species which are potentially not around yet in a given catchment, so ones that we need to be very much on the lookout for and respond rapidly to should they arrive. There's loads of information like that. And the final version is about 92 pages, so much better than my original 800-page mammoth document that was produced. But I was really pleased because even though we managed to cut it down quite massively, we managed to retain a lot of the catchment by catchment data that we'd managed to collate. So what we produced was colour coordinated grids. So black is for species of high concern, red is early detection and eradication, so species that are obviously not well established, so if they do suddenly arrive they need to be responded to very quickly. And then species where obviously long-term management is more realistic, and also where management might not even there might not even be a methodology for management. So this was the kind of colour grid, and we produced these crazy looking charts. So we broke the region down into catchments with species down one side, so that you can actually look at your individual catchment and look at a species and know, kind of relate that back to the level of attention. So is it here yet? Is it a rapid response species? Is it a long-term management plan? That kind of thing. So we were able to retain a lot of that catchment by catchment data, whilst actually reducing the document size into something a bit more manageable. So that was our Southeast RIMP. I would encourage everybody to use the RIMPs. I think they're a great resource. Hopefully they'll be really helpful to people. So do go on to the Rapid Life section of the NNSS website if you haven't already, and have a look at those for your region. They will obviously be updated, as Alexia said, as part of the Afterlife section of Rapid Life. So what more for us in the Medway Valley? Well, our INS work generally continues. So we will continue with our Medway catchment-wide control programme. We will continue to raise awareness of all the invasive species, including things like Asian hornets, which we're trying to get a lot more involved in Kent. We're part of the Asian Hornet Action Team now as well. So we can help sort of tell people yes or no with regards to any identification or photographs that are sent in. We continue to train up volunteers and professionals, and we will continue to work on things like biocontrol as well. So our standard invasive species programme continues. As I say, we've just finished doing a load of giant hogweed work, which has been good. And as for Rapid, well, we were involved with the floating pennywort. That was our small scale control works. And that continues. We're just starting to do that now. It's obviously been difficult and slow to start because of COVID, but we are now starting to get back out on the water and do some work with that. We've written a how-to guide in partnership with the Cam Valley Forum and British Canoeing, so many thanks to those people who helped feed into that. This is mainly for our own volunteers on the Medway, one of which is pictured there. That's Ian, one of our fantastic volunteers. He's been brilliant at helping to remove some of the floating pennywort and to reporting where it is. So we've written this document as kind of like the end of the rapid involvement with the small scale work for our own volunteers, but obviously it can be free to use if anyone else wants it. And we are promoting the RIMPs as well, obviously, as I've just said. So the Southeast RIMP was recently quoted in the Maidstone Biodiversity Strategy document that's going live soon, we hope. So we are promoting it and we are encouraging people to use it, so that's great. So we will continue to do that, and we will continue to do our other RIMS work as well. And that's it from me. Any questions?
Brilliant. Thanks very much, Andrea. That was really interesting. 800 pages was quite long. I was amazed how much detail you produced, but it was all very useful stuff, but obviously nice to have something slightly more succinct. Yeah. One person asked you, although this might be more of a question for me, can I ask why the Thames tributaries, such as the Loddon, Wey, Kennet, etc., were in the Midlands RIMP and not the Southeast?
As far as I can't remember why the regions were the shape they were. I think it was something to do with how the catchments.
Yeah, I think so this is a bit before I started the job and took it on, but I believe that it was based on some Environment Agency catchments or something like that. Yeah, catchment areas.
Not a shape, because part of if you're used to kind of looking at sort of counties, part of Surrey was in the Southeast RIMP and parts in the Midland, and the same with parts of London. It was kind of strange how it was broke up, but it was something to do with the layout of catchments, I believe. You'd have to ask
I think originally we'd wanted to have smaller regions, but there'd been some kind of issue with that, so that's why it ended up being 5. I suppose it would have been an absolute mission to get even more contractors to cover even more areas. But I'm guessing you must have found that quite a challenge in a way, that you had sort of cut off bits in the middle of counties and things. Yeah,
a little bit. I mean, it was great in terms of liaising with other contractors working in the other regions, because sometimes I suddenly realise I've collated a load of data that actually should go into the Midlands one, or that should actually go into the South West one. So the fact that the people doing each of the individual RIMPs were talking to each other was helpful. That wasn't really an issue.
Someone's saying it sounds like CEH catchments. Yeah, that's entirely possible. I can't remember off the top of my head, but there was definitely some sort of official line drawn around it. I've got a document somewhere with it on. Jen, have you had any questions for Andrea? Oh, you're muted, Jen.
Sorry, I'm just that old chestnut. I'm just scrolling down. There's a lot of no. I think they're all still crayfish ones coming to me. No, I haven't got any for you. I think you've got them all, Alexia.
I've got one more from Valerie saying, do you know, are there how-to documents available for other invasive species?
Well, Rapid Life section of the website, look at those, there's loads of them.
Yeah, if you've got good practice guidance there, yeah.
I mean, the Southeast Rim, when we've talked about individual species and management of that species, we've linked across usually to the good practice guides that have been produced as part of Rapid. So yeah, check out the Rapid Life section of the website. I'm
just checking now out of curiosity, because I know we have about 14, 15 species on there. So those of you who haven't been on our web pages yet, it's www.nonnativespecies.org/rapid. I'm just having a quick skim. So there's American mink, American skunk cabbage, carpet sea squirt, curly waterweed, floating pennywort, although Andrea's also got that covered, probably in more detail than we do. Giant hogweed, Himalayan balsam, Japanese knotweed, New Zealand pygmyweed, Pacific oyster, parrot's feather, piri burr, slipper limpet, water primrose, zebra mussel, and terrapin. Although obviously in a lot of these cases, the management is somewhat limited and it's more down to prevention than anything else.
Our floating pennywort guide is mainly as well to do with volunteers from canoe, kayak, so because that's kind of where we've started to move towards, how we've moved towards doing it. So we want to give our volunteers a lot more power to do their bit, whereas previously we've kind of gone, don't touch it, don't touch it. This year we're really trying to empower the volunteers and help them to help us. But we wanted to provide some sort of training, some sort of guide for them. So
yeah, that makes sense.
Obviously, it gives an introduction to floating pennywort and talks about biosecurity in depth and stuff like that. But it's mainly written for someone that might be in a canoe who might want to do some work. But if you want to.
Okay, well, I think we best move on to the next speaker because I'm kind of aware that time is ticking away. Oh, my video's gone off. Didn't notice that. Oh, hi. Thank you very much, Andrea. That was really interesting. If we can move on to Nicola Morris from Southwest Lake Trucks and Kate Hills from Southwest Water, who are going to talk a bit about the AQUA scheme.
Okay, thank you, Alexia. Can you hear me okay?
Yep, can hear you just fine.
Right, so, thank you so much for asking me to speak today. I'm going to be delivering the first part of this and I'm going to hand over to my colleague, Kate Hills. So firstly, thank you to everybody involved in the whole of the project, really. It's been a really interesting day today and the project has been absolutely fantastic. And the other thing I'd like to mention as well is that I can only speak from a Southwest perspective, but those of you that know me will know that I have a number of different hats. So I am project coordinator for SING, the Community Invasive Non-Native Group based in the Southwest. I also chair the Cornwall Invasive Species Forum and co-chair the Southwest Invasive Species Forum with Kate Hills. So with those things that I'm aware of and also listening to some of the talks today, it's absolutely fantastic to see such a coordinated approach in the Southwest region. So yeah, that's really good. So I'm going to talk to you today about the AQUA Biosecurity Accreditation Scheme, which has been alluded to today already. And this was actually the original brainchild of Jen Nightingale, and she realised that with her work with crayfish, she actually needed to develop the Biosecurity Accreditation Scheme to help to protect the native crayfish. Since that original idea, the project has just grown and grown to the stage where now we're actually working with all aquatic invasive species across all freshwater bodies. And so the idea of this scheme, with my work with as Invasive Species Officer at Southwest Lakes Trust, it's one element of the work that I do there, but it's brilliant to be able to bring some of my real passions together, that of invasive species, biosecurity and citizen science as well. So as has already been mentioned, this is a regional pilot trial. involving a number of organisations. And the scheme itself is run by a steering group, and you've heard from all of us today, and you will hear from Kate later. So that's Jen, Kate, Neil, Alexia, and myself. And as I say, it's open to all water bodies with all waterway users. So we're not just focusing on one stakeholder group, we're actually working with all water users who use the freshwater system. And the idea is not to just increase the biosecurity efforts to combat threats that invasive species pose, but also to preserve native aquatic diversity as well. So any centre that it works towards accreditation and achieves that a level of accreditation demonstrates that they're actively conserving their site to maximise native aquatic diversity. Now, the scheme itself is assessed at 3 different levels, from gold through to silver and bronze. Now, the gold award, as you would expect, is kind of the gold standard, really. It's not an easy level to achieve at all. You have to demonstrate extensive action and evidence that action on biosecurity at the site that you're working at. Through to silver, and then bronze, which is our entry level assessment. Again, it's not easy to achieve. It's not an easy qualification, award rather, to get. But if somebody can get the bronze level, then it does actually provide evidence of commitment to biosecurity, but it also gives peace of mind to the waterway users that are actually using those sites as well. That as landowners and land managers, we are actually committed to protecting the waters. Now, there is a very extensive list of criteria which you need to fulfil in order to achieve these levels. And the steering group has been working on refining these quite extensively. But for each of those levels, gold, silver and bronze, we ask for a biosecurity risk assessment to be carried out on each of those sites. So that's standard across all of those levels. With the recording invasive species at bronze level, we're asking for anecdotal reports to be reported through to local record centres, and I record on an annual basis at least. And obviously any alert species would be, the details of those would be sent straight through to the relevant bodies, including the Secretariat and NSIP as well. And then down to, I won't touch on all of these because I realise that we're short on time today. But for the facilities, we actually ask for no additional criteria for the bronze level, because we're very often aware of the fact that the bronze level lakes and reservoirs are often going to be very small fisheries that maybe wouldn't have the funds to provide facilities. But at silver level, we ask the lake managers to provide some kind of cleaning facilities for people that are using their lakes. And then at gold level, we ask for full washdown facilities, but that could be in the form of a number of things, from angling dip tanks to full boat washdown facilities. And then we obviously ask for information to be provided to members, and for general awareness raising materials to be issued to anybody that visits the lakes as well. In particular, we ask people to support the Invasive Species Week, which is an annual event coordinated by the Secretariat, and it's a really good opportunity to get people engaged in the work that they can help out with, as well as the work that we're doing as well. Now, for events, again, we don't ask for any additional information for the bronze level, because usually due to the size of those sites. But for the silver, for anybody entering a site for events, we insist that all of their equipment is clean. We'd hope that were true at any time, but particularly for events when we have people travelling often from other parts of the world, not just within region. So it's really important that we make sure that we clean everything down before they actually enter the site for events. And then for gold level, we ask people not only to check that the equipment is clean when it's entering the site, but also that facilities are provided for people to do that on site as well. And there are various other reports and checks which need to happen as well. And so there's, as I say, a big long list of things which need to be evidenced in order to achieve accreditation. So where are we at the moment with this scheme? Well, it's a Southwest Regional pilot trial, but we do have 60 lakes in the scheme, and I think that's quite a feat considering the current situation. And we do have 34 ready for accreditation at least, and we're just waiting for a few more things at the other sites, and that all of those will be ready then. It's been quite a difficult task to try and continue with that under the circumstances that we're in, but I think we've done a good job. So well done to everybody in the team. We have site guardians recruited at all of those lakes, and I'll explain a little bit more about those in a minute. We couldn't do the work without them. We also provide training for our colleagues and volunteers. So at South West Lakes Trust, I'm continually pestering my colleagues to ask them to attend training sessions and workshops that I run alongside Kate, but also that we encourage them to use the e-learning on the Secretariat's website. It's a brilliant resource, really, really good. And if you haven't been to have a look at it, I do recommend that you do that. We install signs at all of the lakes that we're accrediting, and we also have leaflets available in the permit huts, where we have permit huts and activity centres. We do have a dedicated web page as well now. We have lots of information on there and links to other resources. Social media posts are regular, and also we're often asked to write editorials for other organisations which are released in the press and through newsletters and on websites. Stakeholder engagement is a key part of the work that I do, not just with this pilot project, but with all of the work that I do with South West Lakes Trust. We run biosecurity workshops, and actually Kate and I ran 5 just last year, at the end of last year, which are very well attended, and it covered the entire region from West Cornwall up to Dartmoor, Exmoor, and Bournemouth. We also have open days. Obviously these things have been called to a halt for now, but will resume again when it's safe to do so. We have presence at those open days. We also run evening talks and training sessions for external organisations as well. And Kate's going to talk a little bit about the external validation of the assessment, because it would all be very well for us to say that we'd achieved a certain level of biosecurity, but obviously we need to have some checks in that to make sure that we are achieving the correct level. So we support and promote the Check Clean Dry campaign extensively, because it's one of the most simple and effective forms of biosecurity. So these 2 signs are at one of our lakes in the Bournemouth area. They are the paddle sports signs. So we have signs for paddle sports, sailing, angling, both coarse and game fishing. And also there's a general sign which is more suitable for sites where perhaps we have hikers but not much other activity going on, but it's still important that they understand about biosecurity. So at this particular site, we have the signs at the car park on the entrance where they would take the kayaks into the site, and then on the slipway as well. We have them at all of the angling entrances at all of our sites. And also we make sure that people are very much aware of their obligation to carry out biosecurity by placing them at strategic locations. So the picture on the left at the bottom there is a sign that we've placed at the permit hut, one of our lakes in Cornwall. So anybody that goes to buy a day ticket would have to go into that hut and buy their day ticket. Anybody buying online, and at the moment all of our permits and season tickets are online and also self-launch for water sports, but they have to tick a box online and read the information to say that they agree to carry out biosecurity. But in this instance they must, if anybody buying on site would have to go into the permit hut. But then also on the way out we put another sign up that reminds them before they leave the site that they should clean and dry their equipment. We also have a number of wilderness fisheries, so the people that fish at these sites don't have to buy a permit from us, they just need to turn up with their rod licence, the EA rod licence and their equipment. But it's just as important that these occasional anglers are aware of the fact that they need to carry out biosecurity too. And now, most importantly, this is really a shout out to our site guardians. These are volunteer site guardians who are very much part of this scheme. And these people are our eyes and ears on the ground at all of our lakes. They're the experts at the lakes because they're there so often. I think some of them have actually moved into those sites because they're there so frequently. These are volunteers. Some of them are bailiffs, like Dave in the picture there. He's one of our site guardians at one of our larger lakes in Cornwall. And we make sure that they're familiar with biosecurity procedures themselves, such as clean dry. Then they encourage others to take part in proper biosecurity as well, so other anglers, visitors to the site. We give them full ID and biosecurity training as part of their volunteer work. They carry out regular monitoring at the lake, and they also check all the signage that we've put up as well to make sure that it's all in place and undamaged. and that posters in notice boards are still legible and leaflet stocks are still up to date, so we can then go and replenish if necessary. In cases where we've got angling dip tanks, for example, they also maintain those biosecurity facilities on our behalf. We do have a large catchment area and can't get round to all of those places on a daily basis, so they manage those on our behalf. And then they report any concerns to us immediately, but also as a minimum, every quarter, they then send a form through to us. The form's been developed by one of the volunteers who was a retired auditor. So we've got some real experts within our volunteer base as well. And it's just a really good system. We build up really good relationships with the volunteers, and really can't thank them enough. A lot of these volunteers are also retired industry professionals. So they're not just volunteers who want something interesting to do. These are genuine experts that now have a little bit of extra time on their hands. So yes, it's a really great addition to this scheme. So I think that's it for me, and I'm going to hand over now to Kate.
Thanks, Nicola. So the original aim of AQUA was to protect native biodiversity, but water bodies are much more than just a wildlife haven. They support a range of recreational opportunities, for example, angling, and fishermen don't like crayfish, and you might find that they don't want to restock their fishing lake or pay their licence fees to fish a lake where there are negative impacts from crayfish. Windsurfers can hit free-floating mats of crassula at speed, so there's a health and safety implication there, as there is for people walking around the water bodies like families and the little girl with the giant hogweed blisters show. Next slide, please, Nicola. As a water company, we have obligations to provide a drinking water supply, and invasives can impact on that. They can affect the water quality and the transfer of raw water by blocking up pipes, outtakes or screens. And the picture on the right shows a large mat of crassula around the edge of one reservoir, where it's estimated we have 33 hectares of crassula at one site, which is one of the reasons we support working with CABI. Next slide, please. So in January, in the good old days when you could meet up with people, we had a water company invasives forum and we discussed this AQUA pilot project. It was a good start. They said it was a sensible approach. It was agreed it demonstrates change. So the data collection provides an evidence trail that can be used throughout the business to show risk at various levels. And it also shows the level of investment and capital that you're putting into it. We discussed effort versus benefit and decided the rewards were bigger. You're capturing information in individual site managers' heads that may not be accessible. And if you've got a mobile staff or furloughed staff, it's down on paper too. It's a brilliant way to engage with site guardians, as Nicola has said, who are so keen to learn more about invasives and to get training on it and how to protect their sites. And the real benefit, well, that's not so tangible, but you're preventing the introduction of invasives to your site or spread from it. We talked about benchmarking. It's good internally within the business and between different organisations. It's useful for setting up licence agreements, and anything that promotes biosecurity and engages stakeholders has got to be a good thing. And as Niall mentioned earlier, the Environmental Audit Committee into invasives, this was one of the key messages from it. Next slide, please. But there were concerns, and one of them was the bronze accreditation. Is that really the highest level some sites could get? People may not be happy with not getting the gold level. So this is something for us to think about. Can we put on the signage that this is the highest level a small or isolated, like a wilderness fishery site has, or a site that doesn't have many people? This might be the best level it can get, and we need to say that on the sign or the website, because it really may not be worth investing any more time or money to do more on it. And also we need to bear in mind, we should also be striving to do more and better. And by sharing experiences with developing technologies, best practice will change and improve. So we need to think about that and review it. The water companies were keen to know that there was interest from other organisations, and particularly two. One of them was the Angling Trust. who are keen to support it and are working to develop it. And the other one was the Environment Agency. What did they make of this scheme? Well, they said they welcome this approach. It could be important, an important way to achieve biosecurity outcomes, and it could be important as setting up a biosecurity measure. So that was positive too. Lots of water companies were interested, and particularly Yorkshire Water expressed an interest. And then another big question, what happens when the rapid funding finishes and we lose our manager, Alexia? Well, we are carrying out an audit, as Nicola said. Yes, and as the Angling Trust and Yorkshire Water expressed an interest in this, we wrote them in as our external auditors, which is great. We do need to review it. Is this going to work? Is it suitable to roll out? How can we improve it? And to do that, I think we need to live with it for a while and develop it. So I see it as a trial over the next year. Can we keep our site guardians interested? Is the signage and approach effective? I know in the Southwest RIMP region, we're keen to try and develop it, and Southwest Water and Bristol Water are keen to put in more sites. And I'm delighted to say that Yorkshire Water has already aligned AQUA with its new business plan. So I think there's a lot of interest in it. But one of the big issues is who's going to manage it. And the Water Forum really felt that the Secretariat were the key people to do this. However, there are already a small team who punch above their weight, so that could be difficult. But as Niall said from the Environmental Audit Committee on Invasives, government has recommended that their funding is increased threefold, that's huge, and that there's an inspectorate, which logically the Secretariat should be involved with. So I think there's a real opportunity there. So my last slide is just to thank everyone that's been involved so far, people along the bottom who've helped set up and run AQUA, people who've put sites in, and some of the people on the higher levels hope to work with you more to develop this. And I look forward to working with a lot more organisations and getting people interested. And if you are interested and want to know more, I suggest you contact Alexia while she's still here. Thank you.
Brilliant. Thank you very much, Kate. And Nicola, I enjoyed that. I had one question for Nicola that's come through. What disinfectants are used in angling dip tanks?
So we're using Virkon Aquatic. It's the only licensed disinfectant that we have available to us at the moment for use near water bodies. I've been talking to Braden, who manufacture that, and we've actually got a new protocol. So the interesting thing is that for lightly soiled equipment, such as paddles and kayaks, so if you're spraying as well as dipping, you only need to use a 0.5% concentration, which actually halves your disinfectant costs, which is always going to be a good thing. So yeah, I'm happy to help anybody if they're writing up protocols. We have our own within South West Lakes Trust that we're using now. Just another thing on disinfectants, it's interesting that I've been preaching about using good biosecurity methods for the 10 years I've been working in invasion biology. And it's always been said to me, oh, well, that's something that you do because you're an environmentalist. And I've been saying, no, it's not. It's something everybody does. As a matter of course, it should be part of everybody's everyday life. It takes, sadly, a global pandemic for everybody to suddenly realise that biosecurity should be something that everybody does do. Through that, we've had absolutely no problem in getting all of our activity centres and all of our staff to take up good biosecurity practice. And I've worked with them so that they're actually using Virkon Aquatic to disinfect all of the equipment that comes out of the lake now. That's from a human health perspective, but also from an invasive species perspective, it's a really good thing as well. So yeah, that's a positive that's come out of a very terrible situation.
Thanks very much, Nicola. Just to add to that, so far, at least through Check, Clean, Dry, the main message is obviously about check, clean and drying it, but disinfecting is obviously useful depending on the situation and the context, especially if you've got issues like disease. So that's something that is definitely worth looking into based on what your site's like, I think. Is there any other questions? Oh, one from someone called Simon Owens. Does Ofwat have INS on its agenda for all GB water co-parties? I'm thinking he's written co-parties.
Well, all water companies.
Oh, companies. Sorry. Yeah. There you go.
Yeah, sorry. All water companies have got water industry environment plan objectives on invasives. So it's very positive. A lot of us are pooling together to support coalition approaches. So it's about a dozen of us who are sponsoring NAL and Secretariat to develop Check Clean Dry on that side. Through Yorkshire Water, there's a coalition working to look at raw water transfers and implications. So yes, we do. It's on the agenda very much.
Okay. Are there any other questions? Jen, have you got any?
No, I haven't got any.
If anyone else has anything, please do message me or Jen, or otherwise message Nicola and Kate directly. So I think we'll take a comfort break for 15 minutes. So if we all come back at 3 o'clock, that gives people time to just, go to the loo and get a coffee and whatever else they need to do. And then we'll be finishing up with a talk on biocontrol by Cabby, which is definitely going to be super interesting. And also obviously summing up the conference. And then we have an optional film on crayfish conservation. I appreciate some people might need to get off or have other responsibilities, so you can, watch that if you wish. Oh, I've just got one more question that's come through. If you're using disinfectants, well, no, it's more of a statement. If you're using disinfectants, you need to make sure the equipment and footwear, etc., is completely dry before it is next used, as it can poison aquatic life. Yeah, again, I would say I think it's important to really research into what you're using and where you're using it, and not use it kind of willy-nilly.
I can just have a little bit of input into that. So Burcon Aquatic is the only licensed disinfectant to use near water bodies, because it's actually used as a treatment for treating fish as well. So it's a useful chemical to have. That said, we are constructing dip tanks near the water body, so they're easy for the anglers to use, but away from the water body with a full soak away system, so that nothing is then going directly back into the water body. But you're right, it's a really important consideration to make, and just to make sure that you are using a licensed disinfectant suitable for using near waterways. And as I say, Burcon Aquatic is the only one, as far as I'm aware, that is licensed at the moment.
And yeah, obviously follow the guidance and the instructions. And Neil Green's just mentioned as well, hot water treatment is well worth knowing about too. If anyone's interested in looking about the sort of different efficacy of different treatments and things like that, I would recommend speaking to Stephanie Bradbeer, who is attending today, or private messaging her, because she has a lot of knowledge on that sort of thing. So I think we'll take a break now. If everyone wants to come back at 3 p.m, that would be great. Thanks very much for the talk so far, guys. But I think if you want to start, Kate or Corin, whoever's going first.
Corin has the pleasure of going first. When
Oh, lucky Corin. I hope you've got like some plants to wave around to compete with Jen.
Unfortunately not.
Does that look about right?
Yeah, that looks great.
Super. Right. So yes, shall I go? Are we good?
Yeah, by all means. Oh, sorry, I should probably introduce you. My bad. So we're lucky enough to be partnered up with Cabby, who a lot of you will already have heard of, for the Rapid Life project, and they're helping to steer the biocontrol work, which is where we're working with biological control agents on Japanese knotweed and Himalayan balsam. Unfortunately, we recently lost one of our most enthusiastic friends, Carol, who was also with Cabby and has been really involved with the Rapid Life project. But I'm sure she'll be pleased to know that where the work is still ongoing. And yeah, I'll let Corin take over from there.
Great, thank you very much for the invitation to talk and for the introduction. So yeah, so I'm Corin Pratt. I'll be talking about the biological control of Japanese knotweed, and then I'll hand over to Kate Pollard, who'll talk to you about the Himalayan balsam biocontrol. We're both at Cabby. So Cabby is an international not-for-profit organisation that uses scientific expertise in the fields of agriculture and the environment primarily. And our group particularly focuses on invasive species management, particularly biological control of weeds. So just to get basics of the biocontrol of invasive weeds, classical biocontrol in particular. So non-native plant species introduced to new regions without their natural enemies that keep them in check in their native range. So one of the fundamental theories behind biocontrol is that this decrease in regulation by natural enemies allows an increase in the distribution and abundance of the plant in its introduced range. So this is known as the enemy release hypothesis. In addition, resources that could be allocated to defence, where it's being attacked by natural enemies, can then be reallocated to growth and development in the introduced range where its natural enemies are lacking. And this is known as another theory known as the evolution of increased competitive ability. So these a couple of theories that sort of underpin some of the idea of biological control and how it can work. So the idea of biocontrol is that you can use some of the many arthropods and diseases from the native range that may be specific and damaging enough to then be released as biological control agents in the introduced range of the weed. So here on the left, you've just got some of the natural enemies you find on Himalayan balsam in its native range, so its invertebrates and pathogens. And then at the bottom, you can see introduced gorse in New Zealand, where it lacked natural enemies and grew to these huge sizes. And they were actually quite unimpressed with the gorse they came and saw in the UK when they were looking for biocontrol agents. So it was more stunted and more under control from its natural enemies. So classical biological control uses co-evolved, highly specific natural enemies from the area of origin of the plant to provide self-sustaining control in the invaded range. So obviously not all non-native species become invasive, but when they do, classical biocontrol can be implemented. And this is something that's typically implemented at the sort of later stage of invasion. So after something's arrived, established, spread, and then gone on to have significant impacts. And particularly if traditional methods such as mechanical, manual, or chemical control are ineffective or very expensive, then classical biological control is often considered because it's a kind of self-sustaining method of control where you ideally would introduce an agent and then it would multiply and spread and then bring the target under control. So this is the ideal set of events following a successful biocontrol release. So you have weed density starting at the left of the chart. The weed density is fluctuating with seasons at this high level. And at this high level of weed density, you're going to have significant impacts. So this could be ecological, economic, or even health impacts. And then when you release biocontrol agent, so that's the gray line, this rises to a significant density and then starts to bring the weed density down to a lower and lower level, ideally then maintaining it at this low level below a given damage threshold, which again could be ecological, economic, and then maintaining it at this low level. So you're not aiming to get eradication of the target weed, you're just aiming to maintain it at a lower, less damaging level. And it may be that you need to introduce more than one biocontrol agent, or it may be that the biocontrol just makes the target more manageable, so you can use it as part of an integrated technique in combination with traditional methods like manual or chemical control. And by reducing the population density of the plant down to this lower level, it allows recovery of desirable vegetation. So this is typically native species that have been outcompeted by the invasive plant. So at CABI, we're working on a variety of weed biocontrol projects, and several of these focus on Great Britain, and also Europe, where we do have some common weeds and common interests for biocontrol. So we've got a couple of agents for Japanese knotweed. So firstly, the psyllid, which is allowed for release in the UK, in Great Britain. So that's one that we're currently working on. And another one in development is looking at a leaf spot fungus against Japanese knotweed. There's also Himalayan balsam, which Kate will talk about in a short while. Crassula helmsii, which again, we've got permission for release of a mite in Britain. So that's another one that's actively in the field, and we're working on establishment there. Floating pennywort, that's Hydrocotyl ranunculoides. There's a weevil that's very effective, and a second PRA has just been submitted to the government for review to see if it could be released in the near future. So that looks promising. And then there's the Azolla folliculoides, the floating water fern. And here we mass rear a weevil, Stenopelmus rufinasus, which provides extensive control of that weed here. And then one that's just early in development and primarily for continental Europe is Ludwigia. In the UK, we're still managing to contain this plant and eradicate it where possible. But in Europe, it's very extensively spread across France and also now Belgium and Netherlands. So there we're looking at potential biocontrol. So of these, two that qualified and were suitable for rapid life were the Japanese knotweed using a psyllid as biocontrol and Himalayan balsam using the rust fungus. So we have permission to release these agents in Great Britain, and I will start with Japanese knotweed. So Neil's already done a very rigorous introduction to the Japanese knotweed, so you know about its biology. I'm sure you're all aware of it already from natural environment and people doing work in the field. but it can affect urban environments and natural systems. And here you have it growing along a riparian system, and then classic growing through tarmac shot at the bottom. It has great costs associated with its control, and because of this, a biocontrol initiative was set up just before the millennium, and first surveys were conducted in Japan. And this agent, a psyllid Aphelara itadori, was prioritized as a specific and damaging agent from Japan that could be used in the UK. So, a little introduction to the history of Aphelara in the UK and its biocontrol. So the safety testing was very extensive. It had host range testing against 87 European plants and was found to be highly specific to Japanese knotweed. We also demonstrated later on that it had no significant effects on invertebrates in cage studies and in field studies. It had impact, which was demonstrated particularly by one of our PhD students, who demonstrated that height and rhizome weight in the lab and field was reduced for Japanese knotweed when the psyllid was applied. So in 2010, we had permission to release onto isolated non-riparian stands. So this was the first release of a classical biocontrol agent into Europe. So we had to be very careful. There was extensive monitoring. And the idea was you release onto these small stands, and then if anything goes wrong or there's any non-target effect, you can quickly eradicate the populations. However, it wasn't ideal for the psyllids because the stands were quite dry, small, and isolated from one another. So it wasn't great for trying to establish an agent. After we did these large cage studies in 2014 with high densities of psyllids with native invertebrates, we demonstrated that there was no risk to the native invertebrates from the psyllid. And from 2015, we're allowed to release on riparian systems. And increasingly, we did so with local action group and local authority support, and that's in England and Wales. We tried a variety of different methods then along these riparian sites, conducting also autumn releases. We were also able to get some new stock in 2015, which we started using in 2016 to 17. So we had psyllids in stock in quarantine from the early 2000s. So they'd gone through lots and lots of generations in the lab. So we thought we needed to introduce some increased vigor by collecting fresh stocks from the same location in Japan to kind of bring a bit more vigor into our populations. So from 2016, we've been able to also release the newer stock. And we also tried releasing multiple life stages to allow overlapping cohorts of psyllids in the field. And from those releases, we did actually achieve successful overwintering at a number of sites in England and Wales from 2016 to 17, which was very positive. But then in 2017 to 18, we had poor overwintering, which was, we think, largely down to the weather. It was a very harsh winter and it was quite prolonged. So spring seemed to arrive and then suddenly disappeared, and we had a lot more snow and ice. So we think psyllids probably started to emerge in spring and then were killed off as it was extremely cold and all of their food was frozen off. So on that basis, we're still working for consistent field establishment. We know that when we do releases, we do get reproduction in the field. We have seen overwintering, but typically the population size decreases over the season. And we found particularly very hot, dry periods of summer can be quite negative for the psyllid's survival. So if we release when it's very hot and dry, we tend to see quite high mortality in eggs and nymphs, the junior, these sort of younger stages of the psyllid. So from 2017 to 2020, we've been linked with the EU Rapid Life project. So our steering group for the UK Biocontrol Initiative agreed that the Rapid Life targets aligned very much with the UK targets we already had in place, which gave us the opportunity through Rapid Life to work with regional coordinators across the country. So this was England only, who assisted with site selection, conducted releases, and carried out monitoring. So for Japanese knotweed, we were looking for 3 or 4 sites per region across 5 regions spanning just England. And the license, as mentioned, allows us to now to release onto riparian sites. So we retained any sites that we already had where we were able and renewed permissions to release there. And then the regional coordinators also secured additional new sites. In total, we had 14 sites in England. None of those were in the east, where they had quite active Japanese knotweed control measures in place already, which is a positive thing. But here we prioritized additional Himalayan balsam biocontrol work. So we did, through Rapid Life, we did the psyllid releases in 2018. So we did mass rearing and release, and then started monitoring in 2018, and that continues through to 2020. So as mentioned, the releases and monitoring were conducted by the regional coordinators, and CABI provided some field support, protocols, and some oversight and checking of traps and that kind of thing. and we carried out 3 releases per site, one in early summer, then late summer, and then one in autumn. And the aim of this within this relatively confined window was just to improve establishment. We're not expecting impact in this period. We need to get better establishment with impact a longer term target. So here's just a few photos of the field and some of the methods used for release. So in 2018, we released over 80,000 psyllids. We used degree days to predict emergence. So we mass rear these psyllids inside controlled temperature rooms, and then we're able to ship them in the post. You can see in the bottom left corner, there's a bread bagged Japanese knotweed with psyllids on there. So just as psyllids are about to emerge, we would put them in the post and send them directly to our regional coordinators, who could then release them straight into the field without us having to drive thousands of miles around the country all summer long. So it worked out to be quite an effective method. We had different approaches to release on riparian systems. We did sleeving of plants. We used cutting of plants to provide some young shoots, because there was some demonstration of preference from adult psyllids for feeding on younger, softer shoots, and potentially for ovipositing as well. And yeah, at the bottom right, that's just for one of the local sites we did drive to and then released ourselves, because that was close to Cabby. So unfortunately, 2018 to 19, the field results following the first release, we had poor survival. Again, this happened to be at the start of a very long, hot, dry period. So this, as mentioned, isn't great for the juvenile stages of the psyllid. They prefer higher humidity. We did have higher numbers of psyllids inside some sleeves, but it was variable. And then autumn monitoring indicated quite low persistence of populations, except for one site close to Cabby where we'd put out young boxed plants. The adults seem to move straight onto the young box plants rather than moving onto the tougher, taller, old leathery plants. So again, it's showing this sort of preference for younger, softer plant material. We conducted overwintering releases at that time to try and bolster populations. So here we use an overwintering morph of the psyllid. So we use temperature and day length reductions to trigger the psyllid to produce this darker morph, which will then go and find a local conifer, nestle away for the winter, and then come back out in spring to carry on feeding in spring. We did have some overwintering into 2019, but the numbers were very low, and this was just at sites, a couple of sites in the southwest, one site in the Midlands, and one site in the southeast, the site close to Cabby. And generally, we had very low psyllid recovery through 2019. We don't expect to see every single psyllid in the field. We're just monitoring within the release stand, which is surrounded by other knotweed plants. So there could have been more dispersal and more survival, but still with monitoring and traps, we'd hope to have seen higher numbers. So it's certainly not a large population survival, we don't think. So on that basis, we think establishment is likely to be too low for population persistence. We are going ahead with spring 2020 monitoring. Spring obviously has been shuffled on a little bit with COVID-19 as everything, but we've already had some traps in from people who have been able to go back out to the field. So we hope to assess those traps to see if we have had any population persistence of the psyllid. But for now, it certainly seems that psyllid density is too low for any impact on the plant. So in parallel with this, we looked at climate. So surveys originally focused on Kyushu, which is the island to the south of Japan, as this was the best plant type match to the UK. So the plant in Kyushu was the closest one to the UK as far as we could deter. However, climatically, this isn't an ideal location to collect from because it's more subtropical in the south of Japan, and obviously it's not subtropical in the UK. The psyllids were collected at altitude up the mountains, so the conditions were a lot closer to the UK than the general subtropical region. But based on the results we were getting, very cold winters and hot, dry summers, as well as low humidity in the lab causing poor psyllid performance, we are aware that climate is likely to be very important for the psyllid. So on that basis, we used Climex to find out where in Japan we will likely get a better climatically matched psyllid population that we could use in the UK. So we planned a new survey in 2019, which was approved by our steering committee. And then it was funded by a small consortium of donors to do the work. We then conducted the survey with the aim of sourcing climate matched damaging lines of the psyllid. So we already had Gunma Prefecture flagged from previous surveys as we'd seen significant damage on Japanese knotweed here. And in Murakami, we collected psyllid also, which was further north. So there was extensive leaf curling observed in the field, and it's now been seen in the lab also. We know that our cultures are pathogen and parasitoid free, and we've done molecular testing to ensure this. So we think it's just direct feeding damage from the psyllid, which is a promising impact. So the next steps will be to conduct further host range testing, and then presuming the host range testing continues as so far, which is essentially very, very similar to the psyllid we collected originally, then subject to approval by DEFRA, we may be able to release one of these psyllid lines or both psyllid lines in 2020 to 2021. So this is likely to fall outside the Rapid Life timeframe, but obviously if this is legacy of Rapid Life, it's all continuation of the research findings that we've had through Rapid Life. And it could also be part of an Afterlife piece of work. So at this point, I will now pass over to Kate to talk about Himalayan balsam.
Thank you, Corinne. Let's see if I can smoothly switch to sharing my screen.
Sorry, I need to, yeah.
Is that okay? Can everyone see it? Yeah.
I think you just need to, yeah, there you go.
Okay, good. So as Corinne mentioned, the biocontrol against Himalayan balsam focus on using a rust fungus, Puccinia comoravii varglanduliferae. And to date, we have 2 strains of the rust released. There's one from India and one from Pakistan. So after appropriate safety testing and government ministerial approval, the rust was first released in 2014. And since then, it's been released at 47 different sites across 19 counties in England and Wales. And this year, we were quite fortunate enough to be able to still continue with our release of the rust into Scotland for the first time. And that's by working with collaborators with the Tweed Forum. So our initial releases found that there's much variation in the susceptibility of Himalayan balsam to the 2 rust strains, as you can see by the pictures at the bottom here. So we had sites where you didn't get any infection at all, but also sites where infection was really low, as the picture on the left here with really small infection, small pustules, or on the right where there were much better, bigger pustules. So this led us to believe that there's much more genetic variation in the populations. So we conducted a molecular analysis, which found that there were at least 3 different weed biotypes in the UK that differed in their susceptibility of the rust to the rust. And we matched these biotypes with populations in the native range, for which we've been able to pinpoint a region in particular in Kashmir, where we are working with in-country collaborators to try and identify new rust isolates that would be potentially more compatible with the UK populations. So this is the rust fungus. It has a 2-pronged attack. It infects the leaves during the summertime, which it forms these pustules on the lower leaf surface that draw nutrients away from the plant, so decreases photosynthesis, and as an impact, you see plants tend to have a smaller biomass and produce fewer seeds. It also infects in the spring, where it causes seedlings to become etiolated and warped, as you can see on the picture on the right here, rendering them susceptible to secondary infection by the pathogens and diseases. So a recent studies found that this stage can cause up to 70% mortality of the seedlings in an area, so it can be quite damaging if it gets established. So in 2017, we began working with Rapid Life and regional coordinators, and the aim was to release the rust at 3 to 5 sites per each of the 5 regions, with assistance from the regional coordinators. So they collected seeds from 38 different sites across England, and for us to be able to test susceptibility, we have to inoculate plants and manually infect them with the 2 different strains of the rust to determine which strain is the most pathogenic and virulent. So unfortunately, 20 of these populations were not compatible with either rust strain, so they didn't get infected, and these would be candidates for a new rust strain, ideally from Kashmir if we were able to get one. So out of the 18 that were susceptible, the susceptibility varies. It's on a continuum, so you get some which again have smaller pustules and some which have much larger pustules. So we decided to only release at the ones which had the best infection. So the rust release is actually done by we mix the spores, the brown spores that you saw earlier, into a solution with water and a surfactant called tween, and it's just sprayed manually onto the lower leaf surface using a handheld sprayer. So working with regional coordinators, CABI staff travel to the sites, we train them in the rust release monitoring and protocol, and then they release the rest 3 times in the growing season in June, July and August. So working with Rapid Life, we've been able to release the rest at additional sites across England, which we wouldn't normally have been able to have released at ourselves. So it's been quite a successful result of the project. So in 2018, we released the rest at 11 sites. So 4 of these were our existing rest release sites, one of which unfortunately was cut back by an enthusiastic person trying to control balsam, and 7 of which were new Rapid Life sites. So in total, there was 2 in the north, 5 in the Midlands and 3 in the east. So unfortunately, we found that in particular populations in the southwest and southeast were resistant to the rest. And these are, as we mentioned before, the targets for new rest strains. So generally the rest performed really well in 2018, apart from at 2 sites. So it was Kingsbury Waters and Lady Walk. And these sites we think the rest didn't do very well at was because of more environmental conditions. So the rest doesn't perform particularly well under areas of high shade. There aren't enough nutrients available in the plant to support the development of the rest. So yeah, that's why we suspect these did not do particularly well. The rest, as you can see, it infects the leaves and it persists over winter on the leaf litter where it reinfects in the spring. So we examined all the rest release sites for overwintering and persistence, and we were quite pleased to find that the rest had overwintered at 8 of these. So it was 10 in the end release sites, 5 of which were Rapid Life sites. The 3 sites at the right are our existing sites, and as you can see here, these have a slightly higher proportion of seedlings were infected with the rust. And this is because it's been at the site for 3 years without having supplementary rust released there. So we believe that it's because the rust has established and adapted to UK environmental conditions. So we expect this to kind of happen at the same site. So in 2019, we released an additional vial of the rest at these sites where the rest had overwintered. And we also released the rest at 5 additional Rapid Life sites. So 4 of which were in the Midlands and 1 in the east. Unfortunately, there was another east site that was identified, but we weren't available. We didn't get landowners' permission to be in time for the rest release to take place. So again, we saw really good infection on all the leaves, and the rest was found to be releasing. The rest is starting to spread further from the original rest release patch. So at these newer sites, we found spread up to 15 to 20 metres from the release patch, in comparison to our pre-existing sites where the rest has been for 3 years. We've seen spread from the release patch up to 80 metres. So the rest is adapting to, as we said before, adapting to the environmental conditions. Unfortunately, this year, due to coronavirus, it's not been possible to check the overwintering to see whether the infection is still there at these sites this year. However, regional coordinators will be going back to assess the sites in July and look for the leaf infection on the left here to see whether it's still there. So in summary, the Rapid Life project has increased the number and the distribution of the rust increased the number of sites and the distribution of the rust across the UK. So a total of 11 sites were selected for releasing the rest, and these were mainly in the North, Midlands and the East of England. So good infection was recorded at 9 of these release sites, yet we cannot fully determine whether the rest is established yet because of coronavirus. As mentioned previously, the rest is showing adaptation to the UK field conditions, but as Corey mentioned earlier, the impacts of the biocontrol are long-term, so we're not yet seeing major differences in population density, and these may take 5 to 10 years before they can be seen. The focus of CABI's work now in the future is trying to identify new rust isolates for these resistant populations, in particular those in the southwest and the southeast regions. And yeah, we hope to be able to do that soon. So yeah, as Alexia mentioned earlier, yeah, I want to take this opportunity to mention Carol Ellison, who recently passed away from a prolonged illness. For those who know Carol know that she's a very passionate and dedicated scientist and friend to many people. and she had vast experience in biological control across the globe. She's been instrumental to many of our UK weed biocontrol projects, in particular the Himalayan balsam project, for which she was involved in the original natural enemy surveys and throughout the whole of the project. So into the releases that you can see her here digging up balsam in a patch. So the balsam project and the knotweed project has had quite a large number of collaborators and sponsors and donors, so who I would like to thank. And also I'd like to thank the regional coordinators. So we had Neil, Tarek, John, Andrea, John and Joe and Martin for their assistance during the project. And thank you for listening. That's it.
Brilliant. Thanks very much, Corinne and Kate. So I've got a few questions that have come through. Let's just see if I can get my chat thing to be bigger. Okay, so you have quite a lot of questions. Question on knotweed. Would it be possible to have a psyllid or psyllidia identification sheet plus some images of the damage caused, so as a contractor who have sites extensively across the southwest, we can feedback any observations of establishment in the future?
Yeah, I could happily share those things. At the moment, we do have the monitoring sites that where we've done releases, we expect will be the ones where it's most likely that we would find the psyllid and see any damage. And as mentioned, the population persistence is relatively low at those, and the population density is low. But certainly, if you're monitoring knotweed generally, I'd be happy to provide some images of the psyllid and the type of damage that we see in the lab.
Great. If anyone wants me to put them in touch with Corinne, just let me know. Are Cabby, and there's another question, are Cabby still active with Himalayan balsam in Wales?
So at the moment we don't have any rest release. Oh no, we do. Sorry, we do. We've got a couple of two release sites in Wales, and we are talking to other people who are interested in releasing the rest there. So the way we're kind of going at the moment is working through local action groups who are a bit like through the Rapid Life. We are providing the rest to them, and they're releasing the rest and monitoring for us. So yeah, we have got some along the Dee river at the moment.
Great, thanks. Another question for both of you, I think. How far can the rust and psyllid potentially spread by themselves?
Well, the psyllid is potentially kilometres. It's just a case of, we basically have sites all around the country, which is ideal because you have a number of points from which they can kind of spread, with the idea that eventually you'd cover the whole country. But it depends. When they're in higher density, they're probably more likely to disperse, as with other types of insects. When you get very high density, they tend to move away from that stand into other stands. And it depends on the kind of corridors. So now we're working on riparian systems. They could use these as corridors of spread. But it could potentially spread quite quickly, but won't be likely to spread at such a rate until there's a much higher density of the psyllid in its sort of founding populations, I don't think.
And then,
oh, sorry, you go.
I was just going to say it's similar for the rest. We expect once it builds up to a high enough level that it should be able to spread, hundreds of kilometers. There are examples of biocontrol where it does do, like over 100 kilometers. Is
there like a specific tipping point for when it gets over to that capacity?
Yeah, so we're not really sure. So this is the first time that a fungus has been released in the UK. So yeah, we're kind of trying to monitor and observe it. But the spores that you see on the leaf, they do spread in the wind. So they spread in the wind currents. So.
Okay, here's one for Corinne. Is it logistically viable or cost effective to consider an option for landowners to be able to annually release psyllids if overwintering proves not to be possible?
I don't think so. That's the sort of thing that we do with the Azolla weevil, which is very damaging in a very short period of time. So in 8 to 12 weeks, if we release a given number of seed population of weevils onto a length of canal, for example, they will just feed voraciously and destroy the Azolla. And if in some ways they're too effective, they eat all of the Azolla and then they have to fly away because they've no food left. Whereas the psyllid, as a sap sucker on a very large plant with a lot of resources, the idea would be that it would be a much slower process. So you'd ideally get establishment of populations on a stand, and then they build up to many thousands, and they gradually deplete the knotweed and reduce the sort of resource that's being sent back down to the rhizome. But that would be a much more gradual process, whereas the Azolla is very voracious and it directly destroys the food. So I think that that approach works for some agents, but I don't think the psyllid would be one of them. I think that needs a longer term, large scale establishment and gradual sort of depletion of knotweed reserves.
Okay, that's a good answer. Here's one that I think you guys probably get all the time for both of you. Are you confident that the psyllid or rust fungus won't adapt and predate on any natives?
Do you want to go, Kate? Or?
Yeah, so for the rust fungus, yeah, we are confident. We do stringent host range testing to ensure that it won't infect any native species. So for the Himalayan balsam, it was 75 test plant species, so it includes those that are most closely related. And for the rest to actually infect, sorry. It's thinking of a completely different question that that comes from that one normally. Yeah, so we didn't find that it could infect any other species under optimal conditions for infection. So it's yeah, probably it's unlikely to infect anything in the UK.
And just from a sort of case history, it's extremely rare to see these big jumps. We're working specifically with closely co-evolved, highly specific agents. So the first thing we do is rule out probably 99% of potential agents because they're not specific enough, they're generalist or they're slightly polyphagous, whereas we're basically selecting from the native range the ones that are most specific and are therefore the most closely co-evolved with the target. So of all the insects likely to jump post, they're very low, or the same for pathogens as well. It's a very low risk, and it's not been seen as biocontrol has been practiced for over a century, particularly in places like South Africa, and then more recently Australia, New Zealand, America, and you extremely rarely would get non-target feeding that hadn't been predicted by the scientific study.
Oh, one more question from Tim Agins. Question: zero risk probably doesn't exist. Are risk assessment outcomes for biocontrol agents judged relative to the risk/damages caused by the host IAS? How is the UK government deciding to release or not?
Well, we go through regulation from DEFRA, so it's always their decision, and they have a very thorough process for vetting things. We do the pest risk analysis, and it's thoroughly reviewed by multiple parties, and it's in the end it's their decision. We present this sort of dossier of evidence, and they decide. We have our levels of confidence, and I think the UK has really made great progress, certainly in a European context, for trying to weigh up the pros and cons of biocontrol. So zero risk probably doesn't exist. Low risk, very low risk is ideal, but there's also obviously you have to weigh up the impacts of the invasive species. So in Europe particularly, there's not been a great deal of progress with biocontrol because lots of people in government level, I think, see biocontrol as a risk rather than a potential to reduce the impact of the invasive species, whereas the UK, I think, takes a more pragmatic approach, which is more like that of New Zealand, South Africa, and such like, where they see the impact of the weed as part of the calculation. So an extremely small risk plus the benefit of reducing the impact of the weed is seen as a positive, rather than purely it has to be zero risk, but there's no going ahead whatsoever. So I think the UK government and steering group have been really good sort of moving that process.
It might be also just worth mentioning, do you want to give people an estimate of the average time it takes for the whole process to carry on? Because I know that takes quite a long time to get permission.
It depends on a variety of things. So there's the sort of off-the-shelf sort of biocontrols where you've had a biocontrol agent that's worked against a target weed in another region, and you can effectively very quickly draw up a host range list for the country you want to introduce that biocontrol for, test the plants, and then release, and that's quite a fast process. Whereas if it's from scratch and you've got to start initially by kind of scoping surveys to the native range, try and work out what you find there, do lots of literature research, speak to experts in the field, and narrow down your candidate agents, do lots of testing and quarantine. As for the psyllid, that can take even a decade. Ideally, it's shorter than that, and it can be much shorter than that, as we're demonstrating now with crassula mite. That was a quicker process, and Himalayan balsam was quicker too. But it can take years. So that's one of the fundamental things. It needs investment at the early stages, but then once it's released and you get establishment and impact, that's kind of perpetual. So the investment is up front, and then you get benefits for years and years into the future.
Great. Do you want to add anything to that, Kate, or are you kind of happy
Yeah,
with
no,
that?
I think Corin's covered it. It's just for the balsam, yeah, the project started in 2006 and we didn't release till 2014. So even that stage was, yeah, it takes time, and it's not until we've made sure under quarantine that things are safe before we can even put an application in for release. So yeah.
There's a quick comment from Dr. John Foster saying, did you say this was the first introduction of a fungus, MAF, kindly allowed introduction of crayfish plague? Oh wow, I can't pronounce that. Aphanomyces astaxi across the country when they sponsored dumping of signal crayfish in British rivers in the late 70s, early 80s.
So it's the first release of a fungal biocontrol agent for control of invasive plants. So it's as a classical biocontrol rather than
very specific. Okay,
yeah.
Jen, has anyone sent you any questions?
No, none.
Fabulous. Well, I think we'll start to wrap up, and then we'll show the crayfish video for those people who would like to see that. Obviously, I know some people have to get off. So I just want to say thank you, each and every one of you, for dialing in today. I know this isn't a very normal situation. I know we're all probably going a bit stir crazy, not being allowed out to do as much field work as we'd like, and being stuck at home and, you know, talking to our family a lot. If we're lucky enough to have family with us. In my case, a lot of the time it's the dog and the boyfriend. But I just wanted to say that this project's been absolutely amazing. I know it's had its bumps in the road, like any project, especially long-term projects, especially complex projects like life projects. But I love how many people it's brought together. And you know, you end up getting a stakeholder from France or Belgium, and you sit them down at a table with someone from say Cornwall, like Trevor Reynolds, and actually turns out they got quite a lot in common, really, and you're facing the same problems, and it just goes to show if you can collectively work together, you can achieve so much more. And thank you very much to all the speakers today. I've already said thank you to a lot of other people, and I haven't got the list in front of me, but of course, a particular thank you to the project partners, Bristol Zoological Society and Natural England. Thank you to the European Commission, who obviously run the LIFE programme. It's a shame we can't apply for more EU LIFE projects. I hope we will be able to participate in some way into the future, perhaps. And yeah, it's been a real privilege to work with the people who've been involved with RAPID, and I hope we can ensure there's a legacy after this project, and then it still has useful tools into the future. I'm just going to share my screen right now, if I can get it to work. Ooh, share, okay. So this is my email address. If anyone wants to email any feedback about the conference, or has any questions, or wants to be put in touch with anybody else, because we haven't done the GDPR stuff, we can't keep your emails after the conference unless you're already on one of our mailing lists. So if you would like to be on our mailing list, feel free to email me, or if you'd like to provide any feedback, the website address there is the one for RAPID, and that will be up on the Secretariat website, which is on nativeSpecies.org, for at least 5 years as part of the RAPID Life project. I'm just going to leave that up for a little bit. And also, again, thank you very much to Mark and Jen for helping to do so much with the conference today and dealing with all the tech issues like me disappearing.
And thank you to Alexia. I feel at this point we're probably giving you a nice bouquet of flowers and say thank you very much for your help and just for steering it and for being a great project manager and all of those great things. And yeah, just I'd like to second that, to say thank you very much to all the delegates for sticking with us. And we won't be offended if you don't watch the Crayfish film.
We're going to count every single one of you, and we're going to email everyone who doesn't want to watch.
Yeah, we would really appreciate your feedback. That would be great. If you could email Alexia, that would be great. We couldn't send a feedback form due to some GDPR issues, so it would be great to have that. Thank you.
That would be really good. And also just one final thing, which I forgot to mention, is that the RAPID Life project has had a temporary extension due to COVID. So even though this is the end of project conference, it's actually going to be continuing on for a few more months until we've tied up all the loose ends. So if there is, anything you want to get in touch about, by all means do. We're going to be, circulating reports which are going to assess how well RAPID's done as a project and what we've achieved. This whole day has been recorded, and their slides are going to be available as well. They'll be on our website, and we'll also circulate them on our LAGS mailing list. I appreciate, again, some of you aren't on our original mailing list, and I haven't got organized with the GDPR, but I can keep your emails after this day. So if you would like me to directly email them to you, please get in touch, and I'll make sure you can see them. Otherwise, look on the RAPID website, which is where they'll be. I'm not quite sure how long it takes to sort the recordings out, Mark, but I can hopefully get it up tomorrow.
Yep, that should be fine.
Okay, great. Right, well, so I guess we'll start with the crayfish film, which is, it's actually pretty good. I've seen it, I quite like it, so I would recommend watching it.
Thank you. And as I say, this was from a while back. Oh, more claps for Alexia, sorry. So this was back in the day from the first phase of the project. So it's a little bit dated, and there's Martin Frayling still part of the Environment Agency. But I just thought it would be quite fun to just a bit of light relief at the end of a quite intense day. So I'll try and share it.
See if it works. All right. Just streaming it.
Oh, we can hear it.
I think that's it, if you want to do full screen. Or do I need to do full screen?
I think I'm on full screen.
Oh, Jen, you need to leave yourself not unmuted, otherwise you can't hear it.
With each species on another for survival. Much is known about those animals frequently found at the surface of the water. What are those found deep below, such as this, the native white-clawed crayfish? White-clawed crayfish are a secretive species, playing an important part in preserving the balance of the food chains of our waterways, feeding on invertebrates, and being a good food source for larger mammals and birds. Once common across Britain, this valuable animal is now endangered, and the reason for the disappearance of the species looks remarkably familiar. At first glance, you would not be blamed for thinking that this animal is one of our elusive white-clawed crayfish. However, you would be mistaken. It may be a crayfish, but this is something else entirely. Signal crayfish. Look how it reaches up, defending itself against a predator above the water. The Southwest Crayfish Project agreed to help me get a closer look. Traps are baited with fish to entice the signal crayfish, with oils tantalizingly seeping out into the water. Signal crayfish are known as a menace to anglers, as they often sneak up to fishing lines and steal the bait right off the hook. This means when we place the traps into the river, we are confident that we will be successful in catching this bold and brassy species. These traps are carefully placed close to a large tree in a deep section of the river. Crayfish tend to be found in crevices underneath rocks or in between tree roots, so this is an ideal place to set our trap. It must be tied securely to the bank to prevent our catch being washed away, and equally important is the care taken to remove the trap on our return some hours later. The trap is untied and carefully lifted out of the water, ensuring that no crayfish escape from the hole in the end of the trap. When the traps are collected some hours later and carefully removed from the river, we are not disappointed. Once a trap set in this particular river would have been filled with native crayfish, but now the system is full of the North American signals, which immediately attempt to escape from our trap. Although they look similar to our native white-clawed crayfish, they are much larger and tend to disrupt the delicate balance of aquatic populations, such as invertebrates and fish within our waterways. They also do incredible damage to the banks and foundations of dams, as they create extensive burrows, which often cause the banks to collapse. Maddie Rees from the Bristol Conservation and Science Foundation and Martin Frayling from the Environment Agency explain how this species has affected our native crayfish populations.
This is a North American signal crayfish, and at first glance they seem to have quite distinctive features, with bright red undersides to their claws and often a white turquoise blue signal spot on the top side of their claws. However, it's a common misconception that they're easy to identify because they actually vary greatly in colour from individual to individual, and so they can be confused with native crayfish, for example, the white-clawed crayfish. They were brought over because of their size, and they grow to about double the size of native crayfish, so they were brought over in the 70s for the fishing industry, and they escaped, as with lots of animals that were brought over in that time period, and because they're bigger and more aggressive than our native crayfish, they've done really, really well. And we've actually lost about 70% of native crayfish in the southwest because of signal crayfish and other non-native crayfish, and also a disease called crayfish plague.
One of the big issues with the Environment Agency is illegal trapping for crayfish. To trap crayfish, you require an authorization from the Environment Agency, otherwise it's illegal. And this must not be confused with a rod license because they are two completely separate entities. In order to get details on these authorizations, you should look up the Environment Agency website or contact the local offices. One of the main reasons why we regulate so tightly the trapping of signal crayfish is for the conservation of our native species, which is greatly endangered, and also to help prevent the spread of crayfish plague.
Crayfish plague is a fast killer of native white-clawed crayfish. As a fungal-like disease, it can potentially infect muddy boots or other equipment, such as angling flies, nets, or traps. Before I could go in search of the endangered white-clawed crayfish, the Southwest Crayfish Project ensured my boots were thoroughly disinfected to make sure there was no chance of plague getting into a waterway with white-clawed crayfish. It can quickly wipe out an entire population. Preventing the spread of this disease is imperative to protect our native crayfish. It is something that everyone can help with by thoroughly drying out footwear after visiting lakes and rivers, as the disease spores need moisture to survive. After seeing signals firsthand, I joined the Southwest Crayfish Project on a translocation. Here, they collect native white-clawed crayfish from wild populations which are at risk of being infiltrated by signal crayfish or by crayfish plague before rehoming them in safe sites, which are called arks. The team work hard to catch enough crayfish to create a new population in an ark site. They tirelessly search by hand in the cold water to catch adults hiding under rocks in the river. In deeper areas, traps are used, catching larger adults which are vital to ensure that the new population have animals which are mature enough to breed. Once all the animals have been collected, they are measured and gender and health checked.
Male 45.2,
Female 33.5 with black.
male 40.3, beginning to white claw. Female 28.2, just starting to glare, just. You can see blueness across here, but when they really get into condition, when they get older, they get white and blue areas down the sides.
The animals are then carefully packed together in a protected cool box by the river. Before being transported by car to a safe new home. On arrival, the crayfish are released into an area of good habitat to to start a brand new life, safe from the dangers threatening their old home. Rehoming these animals may not be enough. I ventured behind the scenes at Bristol Zoo Gardens to see the Southwest Crayfish Project's innovative research and breeding facility. Juveniles are carefully monitored, and research is being undertaken to determine the best methods of rearing these animals from newborns to a size large enough to potentially release them into the wild. Research into behaviour, dietary preference, and environmental parameters is being undertaken to ensure that the captive crayfish have the best chance of successfully breeding within the facility. The crayfish in the facility are checked regularly for any signs of disease. The facility itself acts as an arc site, ensuring there is a population that can not only breed and potentially be used to stop wild populations, but also ensures there is a completely biosecure population removed from the risk of contracting crayfish plague. These juveniles are about 2 years old and are large enough to potentially release into the wild without a risk of other animals predating on them. Crayfish can live for about 12 years. However, they have a number of predators, and the first stage of crayfish life is the most precarious time for them. I was lucky enough to witness some babies hatching while at the facility. Unlike many animals, white-clawed crayfish breed around October or November when the water temperatures drop. The females protectively carry a clutch of eggs under the broad tail all through the cold winter months. During the breeding season, it is important to keep the females in good condition and limit factors that may stress the individual animals. A fast increase or decrease in temperature will result in the female losing her eggs, and they only breed once a year. Plenty of refuges are provided in the tanks for both the mother and also the young. Twin wall plastic and gravel works particularly well as a shelter for newly hatched crayfish. The female moves slowly to ensure that she does not damage her eggs. Look closely, and you can see the repetitive throbbing motion of her eggs. This is caused by some specially designed limbs called swimmerets, which continually brush over the eggs to keep them supported and safe. When the water temperatures warm around May and June, the eggs begin to hatch, and the Southwest Crayfish Project team must check the females regularly. After the eggs hatch, the baby crayfish cling to their mother's tail while she protects them from predators such as fish, which would feast on the tiny newborn babies. After a few days, the babies become stronger and will let go of their mother's tails to crawl away into the dark crevices beneath rocks and under tree roots. As the final baby releases its grip on the mother's tail, a chemical reaction occurs like a switch going on inside the adult female, and her protective behaviour will stop. She will start to hunt for food. If the baby crayfish have not successfully hidden, she will eat them, despite having cared for them over the past 8 months. These babies are the future for this species, and they are smaller than a 5p piece. The conservationists are working hard to protect and conserve this valuable species and ensure that our waterways remain healthy. Join the fight to help our British crayfish be here for our future generations to see in the wild.
That is super cute.
That's my daughter a long time ago. That just shows how long ago that was made, because that's my girl when she was 3 and now she's nearly 13.
Oh wow, that's amazing though.
But it's still, I mean, you know, it still stands today, and the project's still.
Not to say that the process hasn't changed, really, has it?
It hasn't changed at all. But yeah, there we go.
Oh, fantastic.
Some of you stayed with us, so thanks for that.
No, there's still 52 people, so.
Yeah.
Wow, brilliant. Right, well, I think we'll probably best to wind up now then. Everyone can go and take it easy after sitting and staring at a screen all day.